ಶುರು ಮಾಡಿದೆ ಮೇಡಮ್.
Yes, sir.
ಸೊ ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು. ಇಂದಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಡಾಕ್ಟರ್ ಲಕ್ಷ್ಮೀ ಮೂರ್ತಿ ಮೇಡಮ್ ಹಾಗೂ ಡಾಕ್ಟರ್ ಸೌಮ್ಯ ವೆಂಕಟೇಶ್ ಅವರು ಎರಡು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮವನ್ನು ಕೊಡಲಿದ್ದು, ಈ ಒಂದು ವೇದಿಕೆಗೆ ಡಾಕ್ಟರ್ ಲಕ್ಷ್ಮೀ ಮೂರ್ತಿ ಮೇಡಮ್ ಅವರನ್ನ ಸ್ವಾಗತಿಸುತ್ತಾ, ವೆಲ್ಕಮ್ ಟು ಮ್ಯಾಮ್.
Welcome, sir. Welcome to the students and the teachers.
ಹಾಗೂ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಅಧ್ಯಕ್ಷರಾದಂತ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ ಅವರನ್ನು ಸ್ವಾಗತಿಸುತ್ತಾ, ಹಾಗೂ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದಂತ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ ಅವರನ್ನು ಕೂಡ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಸ್ವಾಗತಿಸುತ್ತಾ, ಹಾಗೂ ಹಾಜರಾಗಿರುವ ಹಾಗೂ ಹಾಜರಾಗುತ್ತಿರುವ ನನ್ನ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿಗಳು, ಸಹೋದ್ಯೋಗಿಗಳು ಹಾಗೂ ವಿವೇಕ್ ವೃಂದದವರಿಗೂ ಕೂಡ ಸ್ವಾಗತವನ್ನು ಹೇಳುತ್ತಾ, ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಡಾಕ್ಟರ್ ಲಕ್ಷ್ಮೀಮೂರ್ತಿ ಮೇಡಮ್ ಅವರಿಗೆ ಈ ಒಂದು ವೇದಿಕೆಯನ್ನು ಹಂಚಿಕೊಳ್ಳುವಲ್ಲಿ ಓವರ್ ಟು ಯು ಮ್ಯಾಮ್.
Thank you, sir. Thank you so much, sir. Hello, good afternoon, dear students. So this is a topic that we have chosen. There are two particular experiments in the molecular basis of inheritance chapter of second year biology, whether it is CBSE or pre-university, which has got great importance at three different levels of examinations. One is, whatever two experiments we are going to discuss today, you find that 100% there will be a question in the board examination. So much so that if you check the question papers of the PU board exams over the past 10 years, you will find that most of these examinations have both the experiments given in the same question paper also. If not, one of the two experiments will definitely come in the board examination and it is going to be worth 5 marks. It is a very, very, each of the experiments is very logical. It is interesting for students and it is also accompanied by diagrams, and you can expect these questions even in the board examinations that will happen in March. And apart from that, both these experiments are extremely important because NEET questions are very frequently asked on the concepts either directly related or indirectly related to both these experimental, projects that were done by a team of scientists. And apart from that, the third place where these questions will be asked is going to be at the Karnataka CET, right? So all the best to all the students for the upcoming board examinations as well as the competitive examinations. So there are two experiments which I said we will be discussing today. The first experiment is an experiment that is called as the transduction experiment, or it is familiarly known as Hershey-Chase experiment, right? So what is the background for this experiment? Now, what happens is when you look at two biomolecules, that is proteins and DNA, for many scientists, you notice particularly in the 19th century as well as in the beginning of the 20th century, DNA appeared to be quite a boring molecule because it did not show any kind of activity and it was lying within the nucleus of eukaryotes or inside the cytoplasm of prokaryotes. There was hardly any activity shown except during cell division. Right? So what scientists believed was that DNA was mostly a passive molecule without any apparent role. Whereas most of the scientists, particularly biologists, biochemists, they believed that proteins actually were genetic material. Because when we say, for example, let us say, Yaudaru Vandu recipe book kalli, you find that there will be variety of recipes. ಪ್ರತಿ ರೆಸಿಪಿ ಒಂದೊಂದು ಡಿಶ್ಗೆ ಇಂಪಾರ್ಟೆನ್ಸ್ ಇರುತ್ತೆ. ಸೊ ಅದೇ ತರ what happens is when you look at so many different kinds of functional roles that proteins do. And there is so much of diversity with respect to their structure, with respect to their function, etc. Proteins became one of the favorite candidates to be finally declared as genetic material. Now, the students of second year will be familiar that there were many experiments done. Some were done, intentionally, some were done for some other purpose. For example, you had experiments done by Frederick Griffith, where Griffith was mainly working on studying about the disease development with respect to pneumonia in mammals. We also have Avery, McLeod, and McCarthy, who were able to modify and customize the experiment done by Frederick Griffith. So with a lot of other scientists also who got involved in this process of trying to find out the genetic material, you find that somewhere, though DNA people were curious about as to what is this molecule which is there in every living organism. It seems to be passive, but it is something that is commonly seen. And people were also wondering about DNA, whereas proteins definitely, as I said, were a primary candidate. That is when we have, in the middle of the 20th century, two scientists who worked in the Cold Spring Harbor lab. That is, one scientist is Alfred Hershey, and the other is his colleague Martha Chase. And both these scientists, they conducted an experiment that turned up the avenue for more research on DNA, because it finally, without any kind of doubt, laid to rest the notion that definitely DNA is going to be the genetic material. And that was through their transduction experiment. So today we will be studying about this particular experiment. So the experiment, as I said, is named after the two scientists. We call it as Hershey and Chase experiment. Now, what happened was, even though Avery, McLeod, and McCarthy were able to prove that DNA was the transforming principle and therefore it is going to be the genetic material, the problem that happened was that they were working with living organisms which have diverse biomolecules. Okay, so people started wondering whether the experiment was conducted using the right kind of experimental organism. That is when Hershey and Chase decided to choose an entity. Okay, it's difficult to call it as a biological organism because it is not. We'll call it as a biological entity. So they chose a foolproof entity which could compare only between two biomolecules, that is proteins and DNA, without any kind of confusion thrown in from carbohydrates and lipids and RNA, etc. And that biological entity happens to be bacteriophages. I'm sure the students know right from class 11 that except for some of the coliphages, you find that bacteriophages happen to be DNA viruses that infect bacteria. So you're familiar with them. So they decided to choose the biological entity as bacteriophages. You all are familiar with bacteriophage structure, which you have learnt in the first year biological classification chapter, as well as in second year in microbes and human welfare chapter. So a bacteriophage has got almost like a tadpole-like structure. It shows the presence of a capsid, which is comprised of various types of capsomers. There is going to be a neck-like structure with a collar and a base plate having a number of tail fibers that are present. And that completely makes up the capsid of a bacteriophage. And right inside, it shows the presence of DNA as the as the nucleic acid present inside. Let us not try and use the word that DNA is the genetic material. We'll try to prove what Hershey and Chase were able to prove, right? So what Hershey and Chase did as a preparatory step for their experiment, Hershey and Chase firstly, they took bacteriophages and they were able to culture these bacteriophages on very specific medium. Right now, the students will definitely ask one neat question that can come. Was it possible for Hershey and Chase to directly grow the bacteriophages on an agar medium containing radioactive phosphorus and sulfur? This is a good neat level question that can come. And the answer is going to be no. That is mainly because, or it can be given as an assertion reason question also. And the reason happens to be that bacteriophages, being obligate parasites, definitely require a cellular medium to show reproduction, right? Or any kind of metabolic functioning. So you cannot grow them directly on an agar medium. You will have to make sure that there is the host E. coli bacteria that is present on the agar medium. And that is what we call as a culture medium. So any viruses, when you culture, remember the culture medium should have host cells, right? But we do not directly put it in the paper, but this is something that is implied, and it's definitely a neat level question that you can expect. So what they did was they took two batches of bacteriophages. One batch of bacteriophage which was present was grown on radioactive sulfur, that is S35 medium. Remember, when I say medium, it means cellular medium, that is E. coli cell. And then you find that another set of bacteriophages was cultured on a medium that contained radioactive phosphorus, that is P32. Right? So two batches of these bacteriophages were taken. Now, why are we choosing only these two elements? So in order to understand why these two elements are chosen, I think we can now actually begin the experiment. Now, what was the aim of Hershey and Chase experiment? So the aim of Hershey and Chase experiment was to finally lay to rest the question, which was, are proteins the genetic material or is DNA the genetic material? So the aim was to prove which of these biomolecules is the genetic material. That was the aim of the experiment. Right? And when you are comparing between these two biomolecules, then you obviously have to take the difference that occurs between these two biomolecules, right? Now, what is the chemical difference which exists between these two biomolecules? Obviously, it can't be carbon or hydrogen or oxygen or nitrogen, because both proteins as well as DNA contains CHON. Right? Then what is going to be the difference? So the difference is with respect to these particular elements. That is, all proteins contain sulfur, because you all know that methionine is going to be the first amino acid, and methionine is a sulfur based amino acid. So all proteins contain sulfur, particularly all bacteriophages 100% will contain sulfur. There may be some rare proteins in the living world where the methionine can be replaced or removed, but bacteriophages will 100% contain methionine and cysteine also. So sulfur is definitely present in them. So proteins contain sulfur, but we know that DNA does not have sulfur. Whereas DNA contains phosphorus and proteins do not have phosphorus. So the difference between them chemically, when you look at the elemental level, is going to be that proteins have sulfur and DNA has phosphorus. So therefore, these two elements in their radioactive isotope forms were taken to culture the bacteriophages, which means if you start with the batch which has got sulfur, you find that the capsid becomes radioactive and the DNA inside is going to be non-radioactive. If you take the batch which is grown on phosphorus 32 cellular medium, you find that the capsid remains non-radioactive, whereas DNA is going to be radioactive. So once this is done, now we will begin with the first step of the experiment. So the first step of the experiment that they did was going to be called as infection. So step number one was called as infection. So what does this infection step mean? I think the word is self-explanatory. So what was done? A fresh batch of E. coli was taken, not the ones that were used in the culture medium. Fresh batch of E. coli was taken, and these E. coli cells which are present, right? We'll just draw them as the bacillus structure. These E. coli cells were taken. This batch was infected with the bacteriophages containing radioactive sulfur. Another batch of E. coli cells, completely separate batch, was infected with the bacteriophages that I've drawn on this side. So the first step was infection, right? Which means if we have to draw it separately, we can show it in a very neat manner. Right? So what we find here is going to be this. So one batch of the E. coli cells which were present were allowed to be infected by the bacteriophages which were present in this manner. And this side, you have the E. coli cells in step number 1, that is infection, which were allowed to be infected by the bacteriophages containing S35, right? So this was going to be step 1. Why don't we show the nucleoid or the plasmids of the bacterium? It is only for diagrammatic purpose so that students do not get confused, right? Now going ahead, this was the first step. So we know what is going to happen in infection. In infection, you find that the bacteriophage is going to penetrate the cell envelope of the bacteria and is going to inject the genetic material into the respective host cells, right? How do we do this? We can do it in a Petri dish. A Petri dish is going to be easy for infection. Once infection was done, the next step which took place, second step, so this was the first step which was infection. The second step which took place is going to be this way. So what was done in the second step? Step number 2 is going to be called as blending. So what was done was Hershey and Chase used a centrifugal machine and they wanted to separate the empty capsids from the infected cells. But a centrifugal machine has got a very high RPM and it ended up, not giving them the result that they expected. So therefore, Martha Chase brought her own kitchen blender from her home, and using that, the infected cells with the capsids attached to them were put in the blender and just given one churn. So once they were able to get rotated, what happened was it loosened the capsids from the infected cells. So blending, how are we going to show? Let us say this is the cell from this batch. This is the cell from this batch. These are the E. coli cells that we are looking at. You find that this already has got the genetic material of the bacteriophage. You find that the bacteriophage is going to be loosened completely or is going to be detached completely from the infected cells. Similarly, here also you will find that the capsids are going to become loosened. These capsids which do not have genetic material in them are called as ghosts. GHOSTS. They are called as ghosts. That is, empty capsids are going to be seen, right? Once this happened, this step was called as blending because you just used a home blender in order to separate the empty capsids. Once this was done, now they moved to the third steps. So the third step which took place here was going to be called as centrifugation. So step number 3 was called as centrifugation. I'm sure the students are familiar with this particular, it's a physical way of separating substances based on density. So centrifugation at a certain RPM, you will be able to separate them. So once the test tubes which contained, please remember, these are the test tubes in which you have a suspended turbid solution that is going to be seen. You are unable to see which is going to be having radioactivity. None of them you are able to see with your naked eye. There is just the suspended turbid solution that you have produced after blending, and you know that inside this, not only do you have the E. coli cells that are present with the respective genetic material and the empty capsids, but you are unable to see any of them. It just looks like a turbid solution sitting here. Now, these test tubes were now given a turn in the centrifugal machine. Once that was done, you find that there were neatly two layers that were separated in the test tubes. Now that interpretation is most important here. So you have the test tubes that are present. I'm drawing it big only for effect here. So the test tubes are present and you have two neat layers that are going to be seen. What are the layers that are going to be seen? So you find that one of the test tubes shows the presence of very clear non-radioactive when exposed to let us say an X-ray film. You find there is going to be clear non-radioactive clear fluid present on the top, which we call as the supernatant. This is going to be non-radioactive. And at the bottom, you find that there is going to be a small little solid pellet that is going to be seen. So the pellet which is present is going to be radioactive in nature. That is, you find this is the one that shows radioactivity. What does the supernatant contain? Supernatant will always contain the ghosts, which are the empty capsids. What will the pellet contain? Pellet always contains the cells which show the presence of the genetic material from the bacteriophage. So pellets will always be the cells and supernatant will always be the empty capsids. Even here you are going to get two layers. So there will be two layers that are present. So there is one layer on the top and then at the bottom you will find that there will be pellets present. Okay, so this pellet is going to be non-radioactive and you find that the supernatant is going to be radioactive and supernatant is made up of empty capsids and this is going to be made up of the cells which are present here. Right now, how do we interpret the data? Because the experiment is done, so all we need to do is interpret the data. Let us interpret in this way. If proteins were the genetic material, then proteins should have been inserted into the host cell for necessary reproduction of the viral particles. So if proteins were the genetic material, then protein should have been inserted into the cells, which means beginning from S35, you should have found radioactivity in the pellet, that is in the cells, and not in the supernatant. Absence of radioactivity in the batch beginning from S35 in the pellets, that is in the cells, shows that proteins were never injected into the cells, which means proteins are not the genetic material. So this part of the experiment shows proteins are not genetic material because they did not land up in the cells. If they had landed up in the cells, then in that case, the cell should have been radioactive, but they are not. Whereas here you find that the pellet shows the presence of radioactivity, and where was radioactivity present in this batch? Starting from P32, it was present in the nucleic acid and not in the capsid. Which means if radioactivity is seen in the pellet, that means the cells have radioactivity. That means the cells got injected with the DNA. And what gets injected into the cells? Genetic material. What got injected? DNA, which means DNA is going to be the genetic material. So this batch of the experiment proves that DNA got injected into the cells, and because what gets injected into the cell is genetic material, DNA is the genetic material, right? So this is a simple transduction experiment done by Hershey and Chase. right? So as I said, this particular one, I have tried to split the diagrams into 3 parts because I'm trying to draw it, you are trying to see it online. If you were here directly in the class, in front of me, we could have drawn the entire experiment together as one diagram. But please learn to draw the diagram because this is a short question in the second year board examination along with the explanation. Right. Let us now go to the second experiment that we are going to discuss today. Right. The second experiment came because we have the 2 Nobel laureates, that is Watson and Crick, who were able to give us the double helix model. They had suggested that when they suggested the DNA structure, they suggested that the DNA structure shows the ability to show a type of replication called a semi-conservative replication. Let us try and understand what is meant by semi-conservative replication. ಈಗ ನೀವು ಫೋಟೋಕಾಪಿ ಮಾಡ್ತೀರಲ್ವಾ, ನಿಮ್ದು ಯಾವ್ದಾದ್ರು ಒಂದು ಶೀಟ್ ನ ತಗೊಂಡು ಜೆರಾಕ್ಸ್ ಅಂತೀವಿ, ಜೆರಾಕ್ಸ್ ಅಂದ್ರೆ ಒಟ್ಟು ಫೋಟೋಕಾಪಿ ಮಾಡ್ಕೋತೀರಲ್ವಾ, ಯಾವ್ದಾದ್ರು ಈಗ ನಿಮ್ದು ಟೀಚರ್ಸ್ ಕೊಟ್ಟಿದ್ದು ಶೀಟು ಇಲ್ಲ ನಿಮ್ದು ID ಕಾರ್ಡ್ ಎಲ್ಲ. ಸೊ ಫೋಟೋಕಾಪಿ ಮಾಡ್ಕೊಂಡ್ರೆ ಏನ್ ಮಾಡ್ತೀರಾ, original paper is as it is, ink is as it is on the original paper, ಹೊಸ sheet of paper, ಹೊಸ ಇಂಕು ನಿಮ್ದು ಕಾಪಿಲ್ ಬರುತ್ತೆ. That means the original is conserved as it is. So for example, if this is going to be the original DNA molecule which is present, I am able to make this into 2 types of copies. And what are the 2 copies? The original is retained as it is, and using fresh material, I'm able to get a new copy of DNA. Right? This type of replication method is called as conservative replication method. And many biologists had an idea that DNA replication is going to be conservative in nature. One more type of understanding which was done, or assumption that was done by scientists, of course it was discarded immediately by themselves, was, is it possible that DNA replication is going to be disruptive in nature? Disruptive andreno, for example, ನೀವು ಒಂದು ನಿಮ್ದು ಆಧಾರ್ ಕಾರ್ಡ್ ನ ಲೆಟ್ ಅಸ್ ಸೇ ಫೋಟೋಕಾಪಿ ಮಷೀನ್ ಗೆ ಹಾಕಿದ್ರಿ, ಆ ಕಾರ್ಡು ಪುಟ್ಟ ಪುಟ್ಟ ಪೀಸ್ ಅಲ್ಲಿ ಎಲ್ಲ ಬ್ರೇಕಪ್ ಆಗ್ಬಿಡುತ್ತೆ, ಆಮೇಲೆ ಹೊಸ ಪೇಪರುವೆ ಬ್ರೇಕಪ್ ಆಗತ್ತೆ, ನಿಮ್ಗೆ 2 ಕಾಪಿ ಬರುತ್ತೆ. ಪ್ರತಿ ಕಾಪಿಲುವೆ ಯು ವಿಲ್ ಫೈಂಡ್ ಒಂದೊಂದ್ ಪೀಸು ಹಳೇದು ಒಂದೊಂದ್ ಪೀಸ್ ಹೊಸದು ರ್ಯಾಂಡಮ್ ಆಗಿ ಒಂದು ಮೊಸೈಕ್ ತರ ಪ್ರಿಂಟ್ ಆಗುತ್ತೆ ಅಂತ ಅನ್ಕೊಳ್ಳಿ. ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಡಿಸ್ರಪ್ಟಿವ್ ರೆಪ್ಲಿಕೇಶನ್. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ವಾಟ್ ಎವರ್ DNA is present is going to break up into small pieces in this manner. Once it breaks up into small pieces, we will take fresh nucleotides and then we will be able to join them together. So what happens is you will get DNA which is going to be in this manner, little bit old and little bit new. Right? This method was called as disruptive DNA replication, where every strand has got 1 nucleotide old, 1 nucleotide new, etc. in a random manner. This was immediately discarded by scientists, saying that if we break up DNA in this way, where is our reference to find out how to make a new DNA molecule? Therefore, it was completely discarded by scientists. So the type of replication that was proposed by our Nobel laureates after the double helix model was made was, if this is going to be the DNA mother molecule or parent molecule, then replication is going to be in such a way that this is what is going to happen, right? 2 daughter molecules are created. Each daughter molecule will have 1 old strand and 1 new strand in this manner. Andre photocopying example do, now if we are able to give an analogy is, ನೀವು ಒಂದು ಲೆಟ್ ಅಸ್ ಸೇ ಒಂದು ಕ್ವೆಶ್ಚನ್ ಪೇಪರ್ ನ ಒಂದು ಫೋಟೋ ಕಾಪಿ ಮಾಡ್ಕೊಂಡ್ರೆ, ಒರಿಜಿನಲ್ ಪೇಪರ್ ಇಸ್ ಹಿಯರ್, ಆ ಇಂಕ್ ಎಲ್ಲ ತೆಗೆದು ಹೊಸ ಪೇಪರ್ ಮೇಲ್ ಬರುತ್ತೆ, ಹೊಸ ಇಂಕು ಹಳೆ ಪೇಪರ್ ಮೇಲ್ ಬರುತ್ತೆ. ಸೊ ನಿಮ್ಗೆ 2 ಕಾಪಿ ಸಿಗುತ್ತೆ. ಆ 2 ಕಾಪಿಲಿ ವಾಟ್ ಯು ಫೈಂಡ್ ಇಸ್ old paper, new ink, old ink on new paper. Like that, you find that DNA replication is going to be in such a way that the old and the new strands will join together to form the daughter molecules. And this was, proposed by the Nobel laureates, but proving whether this is how it is going to occur was a challenge for scientists. And among that, you find that 2 groups of scientists started working to find out, to prove what kind of DNA replication is going to occur, whether it is conservative or semi-conservative. Disruptive, all of them knew is not going to make sense. And one group of scientists led by Taylor were able to work on a particular member of Fabaceae called as Vicia faba, that is the faba beans, and they were able to find out whether chromosome duplication is semi-conservative. At the same time, we have 2 scientists called as Meselson and Stahl. These 2 scientists, they performed what is considered to this day as the most elegant experiment ever done in biology. And that particular experiment, using heavy DNA, hybrid DNA, and light DNA, was performed by Meselson and Stahl. Let us see what this experiment is all about. So what Meselson and Stahl did, they took a group of E. coli cells, Escherichia coli cells. They took a group of E. coli cells and they grew these E. coli cells, which are present for many generations. So for many generations, these E. coli cells were grown on heavy NH4Cl. That is ammonium chloride containing N15. This is the heavy isotope of nitrogen. This question is also asked in NEET exam, where you find they will ask you whether N15 is a radioactive isotope or whether it is going to be a heavy isotope. So it is a heavy isotope of nitrogen. So they grew E. coli for many generations on a medium containing NH4Cl with heavy nitrogen as the only source of nitrogen. When they did that, all compounds containing nitrogen, that is DNA, proteins, etc., all the compounds containing nitrogen, they had heavy nitrogen only in them, including DNA. Right? So once this was done, let us see what was done now. So let us consider, once the E. coli cells, they are on a Petri dish, what was done now? So now we start the experiment after the preparatory step. We are going to now take a reading at 0 minutes. What is the 0 minute reading? 0 minute reading is after growing E. coli for many generations in heavy nitrogen NH4Cl. What was done? What was done is a small colony of E. coli was taken out. Once the E. coli colony was taken out, the cells were immediately killed. You can kill them very easily by putting them in a certain salt solution. It is very easy to kill them. So they were immediately taken at a particular point of time and put into a test tube containing a salt solution. What salt solution was preferred by Meselson and Stahl? So they preferred a salt called as cesium chloride. So when you take the E. coli cells and put them into cesium chloride solution, the cells will stop dividing. They cannot divide in that medium. So the cells are killed. Once the cells are killed, what is going to be done? Same way you can even utilize maybe a strong solution of NaCl, etc. Once this is done, DNA was extracted from the cells because we now know that the cells will not divide. They have stopped dividing at that point of time. DNA can be extracted. Once the DNA is extracted, I'm sure the students are familiar with DNA extraction. Once the DNA is extracted, Now what is going to be done? You are now going to centrifuge the DNA which is present at a very high RPM for many hours in cesium chloride solution. This type of centrifugation, which is done at high RPM for many hours, is called as cesium chloride density gradient centrifugation. Of course, all centrifugation is density gradient, but sometimes what happens is if the centrifugal force is for a lesser time or it is at low RPM, then some gross density gradient can be created, but not very fine ones. So multiple hours when you spin them on a centrifugal machine, even small differences in density you can separate. So when they did that at 0 minute, what they found is you find that the centrifugal tube which is present, usually in a centrifugal machine, the centrifugal tubes become almost horizontal. You can draw them horizontal or you can draw them vertical, doesn't matter. So you find that at this point of time, there is one band of DNA which is present at the bottom of the tube. And this particular DNA which is present, we call that as heavy DNA, which is nothing but 15N 15N DNA. What does this 15N 15N DNA mean? It simply means that both the strands of DNA which are present will show only heavy nitrogen containing nitrogenous bases. That's why it is called as 15N 15N DNA. Now, in the Petri dish, you still have some E. coli cells which are present, right? Now, what is going to be done? This one batch of assistants can continue to do this experiment. Now, what happens? What happens is in the same Petri dish, you now take a few of the E. coli cells and you shift them to a Petri dish which contains normal NH4Cl. That is NH4Cl containing N14 nitrogen. So when you shift them, you will shift them and keep them in the new nutrient medium containing normal NH4Cl for 20 minutes. When you keep them in that medium for 20 minutes, what is going to happen? At the 20th minute, again, you are going to arrest the cells. As I said, you take a small colony and you put it in a salt solution, etc., the cells will stop dividing. Then aram se you can extract DNA from them and you spin. When you spin, what is going to happen? You will find that DNA will again occupy a particular gradient, which is going to stabilize it with cesium chloride gradient. That is going to be seen, right? Now, what you notice is in that test tube at the 20th minute reading, you will find if DNA replication was conservative, you would have noticed that this DNA molecule remains as it is, and the new DNA molecule, which I prefer to draw in white, would have been N14, N14. That is, the E. coli cells for 20 minutes, usually 18 minutes time is taken for DNA replication if we assume. Then they would have used normal NH4Cl as a source for making the nitrogenous bases and finally doing the DNA replication. Which means if you're using the new nucleotides to make the new DNA molecule and retain the old one, you should have got two bands as heavy DNA and one light DNA. Right? If DNA replication was conservative, but what Meselson and Stahl were able to show was that this did not happen at the 20-minute reading. What happened in the 20-minute reading was this. They were able to get a test tube which showed that there was a hybrid band that was seen, right? This was hybrid DNA. That is, it showed 15N and 14N type of DNA. It was a hybrid band that was seen. The position was neither light nor heavy. And what was this? This had two molecules here. You can even extract the DNA and wait to find out whether DNA content was double or not. That can be done. And this was able to show two things. That is, the DNA molecule, each molecule had one light strand and one heavy strand, that is one with F15N and one with 14N, which proved that DNA replication is semi-conservative. NEET, they will ask a question. 2022 also, we had a NEET question which says at a particular generation, let us say this is going to be the parental generation. This is generation 1, generation 2, generation 3 as you go. You can ask a question saying in generation 4, how many DNA molecules are going to be light, or how many DNA molecules will be hybrid, etc. Such questions can always come in NEET exam as well as CET exam. Right now, you know that the paper pattern has changed for the PU exam. So even those MCQs can be asked in the board examination section A also, right? Now, if you continue this for another few minutes, let us say you continue to let the E. coli cells, the ones which are intact, not the ones which were killed, if you allow them to continue in the light NH4Cl for another 20 minutes, that is till you reach 40 minutes, you will find then you arrest the cells and you do DNA extraction, you will find that there continues to be one hybrid band which is always present and one band is going to be light. Right? Please remember, between this step and this step, you cannot prove semi-conservative replication. Semi-conservative replication can only be proved between this generation and this generation. Please remember that. So at this point of time, we are going to have, there will be two molecules which are going to be hybrid in nature, right? And there is going to be two molecules here which are going to be having only light DNA in this manner. Between these two, if you see, many students will think, aren't these two conserved here, and am I not making two DNA molecules fresh? No. Okay, for example, this white line is present here, this pink line is present here, this white line is present here, and this pink line is going to be present here. So these two are not conserved there. They are also split because you know the mode of DNA replication, which is semi-conservative. DNA replication is also an important question, but that is not the scope today that we have, right? Okay, so these are the two experiments, that is Meselson and Stahl, to prove semi-conservative DNA replication, and you have Hershey and Chase experiment, which was finally unequivocally, which laid to rest the debate whether proteins or DNA is the genetic material. Okay, over to you, sir. Thank you, students. Over to you, sir.
Okay, thank you, ma'am.
Thank you, sir.
ಸೊ ವಿದ್ಯಾರ್ಥಿಗಳೇ, ಯಾವುದಾದ್ರೂ ಪ್ರಶ್ನೆಗಳಿದ್ರೆ ಇವತ್ತಿನ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದ ಮೇಲೆ ಪ್ಲೀಸ್ ಆಕ್ಸ್ ಟು ಲಕ್ಷ್ಮೀಮೂರ್ತಿ ಮ್ಯಾಮ್.
Yes, sir. And two simple questions. ನಾನು ನಿಮಗೆ ಟೈಪ್ ಮಾಡು ಕಳಿಸ್ತೀನಿ. Students, very important these questions are going to be. ನಿಮಗೆ 1 mark question MCQ ಥರ ಬರ್ಬೋದು. What was the aim of Hershey and Chase experiment? ಅದು MCQ ಥರಾನು ಬರುತ್ತೆ. ಪೇಪರ್ ಪ್ಯಾಟರ್ನ್ ಚೇಂಜ್ ಆಗಿದೆ, ನಿಮಗೆ ಗೊತ್ತಿದೆ. ವಾಟ್ ಇಸ್ ದಿ ಏಮ್ ಆಫ್ Meselson and Stahl experiment? ಇದು ಬರುತ್ತೆ. Or they will say, ಇನ್ನೊಂದು question ಬರ್ಬೋದು MCQ ಏನು ಅಂದ್ರೆ, Meselson and Stahl ಯಾವ type of centrifugation ನ use ಮಾಡಿದ್ರು ಅಂತಾನು MCQ ಬರ್ಬೋದು. That is also there. Plus, ನಾನು ಮುಂಚೆನೆ ಹೇಳ್ದಂಗೆ both these are very important bold type questions for 5 marks including the diagram. ನಾನು ಸರ್ಗೆ questions ನ ಕಳಿಸ್ಕೊಡ್ತೀನಿ, ನೀವು ಬರೆದು ನಂಗೆ ಕಳಿಸಿದ್ರೆ ನಾನು ನಿಮ್ಗೆ ಯಾವ ತರ ನೀವು ಬರ್ದಿದ್ದೀರಾ ಅಂತ I can evaluate. Yes, sir.
Okay, thank you, ma'am. ಯಾವುದೇ questions ಇಲ್ಲ.
Thank you. Thank you, sir.
ಏನಾದ್ರು ಕಳಿಸಿದ್ರೆ later I send through mail.
Definitely, sir. Sure, sir. Sure.
So,
sure, sir.
Delivered a very good, interesting talk on today's topic. So this is very informative to students. ಸೊ ಇದರಿಂದ ಹಲವಾರು ಮಾಹಿತಿಗಳು ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ತಿಳಿಸಿಕೊಟ್ಟಿದ್ದಾರೆ ಮೇಡಮ್ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಿಂದ. ಸೊ ನನ್ನ ವೈಯಕ್ತಿಕವಾಗಿ ಹಾಗೂ ನಮ್ಮ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿಯ ಪರವಾಗಿ ಧನ್ಯವಾದಗಳು ಮೇಡಮ್.
ಥ್ಯಾಂಕ್ ಯು ಸರ್. ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್ ಸರ್.
ಹಾಗೂ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಹಾಜರಿದ್ದ ನಮ್ಮ ಅಕಾಡೆಮಿ ಅಧ್ಯಕ್ಷರಾದಂತ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ ಕೂಡ ಧನ್ಯವಾದಗಳು ಸರ್. ಹಾಗೂ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನು ಆಯೋಜಿಸಿ ನಮಗೆ ದಿನನಿತ್ಯವೂ ಸಲಹೆ ಸೂಚನೆಗಳನ್ನು ನೀಡುತ್ತಾ ಬರುತ್ತಿರುವ ನಮ್ಮ ಅಕಾಡೆಮಿ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದಂತ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ಗೂ ಕೂಡ ನನ್ನ ಧನ್ಯವಾದಗಳು ಸರ್. ಹಾಗೂ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಹಾಜರಿದ್ದ ನನ್ನ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿ ವೃಂದದವರಿಗೂ, ವಿವೇಕ್ಸ್ ವೃಂದದವರಿಗೂ ಹಾಗೂ ನನ್ನ ಎಲ್ಲ ಸಹೋದ್ಯೋಗಿ ಮಿತ್ರರಿಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತಾ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನು ಮುಗಿಸುತ್ತಾ, ಹಾಗೂ ಇನ್ನು ಕೆಲವೇ ಕ್ಷಣಗಳಲ್ಲಿ ಡಾಕ್ಟರ್ ಸೌಮ್ಯ ವೆಂಕಟೇಶ್ ರವರು ಉಪನ್ಯಾಸ ನೀಡಲಿದ್ದು, ಹಾಜರಾಗಿರುವ ಎಲ್ಲಾ ವಿದ್ಯಾರ್ಥಿಗಳು ಇದೇ ರೀತಿ ಹಾಜರಿದ್ದು ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮವನ್ನು ಕೇಳಬೇಕೆಂದು ಮತ್ತೊಮ್ಮೆ ತಿಳಿಸುತ್ತಿದ್ದೇನೆ. ಥ್ಯಾಂಕ್ ಯು ಮ್ಯಾಮ್.
ಥ್ಯಾಂಕ್ ಯು ಸರ್. ಥ್ಯಾಂಕ್ ಯು.
ಹಲೋ ಮೇಡಮ್. ಸೌಮ್ಯ ಮೇಡಮ್.
Yes, sir.
Audible.
Board is also visible, right?
Yes, audible and visible both are nice.
Yes, sir.
ಸೊ ವಿದ್ಯಾರ್ಥಿಗಳೇ, ಈ ಎರಡನೇ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಡಾಕ್ಟರ್ ಸೌಮ್ಯ ಕುಮಾರಿ ಮೇಡಮ್ ರವರನ್ನು ಸ್ವಾಗತಿಸುತ್ತಿದ್ದೇನೆ. ವೆಲ್ಕಮ್ ಟು ಯು ಮ್ಯಾಮ್.
Thank you, sir.
ಸೊ ಇದೇ ರೀತಿ ನನ್ನ ಎಲ್ಲ ಸಹೋದ್ಯೋಗಿಗಳು ಹಾಗೂ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಅಧ್ಯಕ್ಷರುಗಳಾದಂತ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ ಹಾಗೂ ನಮ್ಮ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿ ರಮೇಶ್ ಸರ್ನು ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ನನ್ನ ಎಲ್ಲ ಸಹೋದ್ಯೋಗಿ ಮಿತ್ರರು, ವಿದ್ಯಾರ್ಥಿಗಳು ಹಾಗೂ ವಿವೇಕ್ಸ್ ವೃಂದದವರನ್ನು ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನು ಮುನ್ನಡೆಸಲು ಡಾಕ್ಟರ್ ಸೌಮ್ಯ ಕುಮಾರಿ ಅವರಿಗೆ ಓವರ್ ಟು ಯು ಮ್ಯಾಮ್.
Thank you, sir. Good evening, all of you. Good evening, students. Right at this moment, maybe for you a kind of quick revision is important. Keeping your competitive exam as well as board exam like very close to you. I think this particular topic what I have selected, you know, both named reactions I have taken from entire organic chemistry. It happens so ಹೋಗ್ತಾ ಹೇಗಾಗುತ್ತೆ ಅಂದ್ರೆ ಸೆಕೆಂಡ್ ಇಯರ್ ಅಲ್ಲಿ ನಿಮಗೆ you have a part 2 of NCERT completely organic chemistry. In that you have 4 important chapters. ಎಲ್ಲಾದ್ರಲ್ಲೂ ಒಂದಷ್ಟು named reactions ಬರ್ತವೆ. When you look into your board examination paper, already you might have written your preparatories also, model papers ನೋಡ್ತಾ ಇರ್ತೀರಾ. There are a lot of questions asked on especially named reactions. At the same time, if you take up any competitive examination question paper, maybe it is a CET or NEET or JEE mains, usually the questions will be on named reactions. So hagagi, I thought a quick revision of these named reactions will be definitely useful for you at this level. So I'll be giving you only the named reactions of all the 4 important chapters of organic chemistry as much as possible, and I'll be taking you through what type of MCQs will be asked for the competitive exam. So that will be the entire session for today. Okay. So when we talk about named reactions, the first chapter you all learn about haloalkanes and haloarenes, right? So you can start listing out because that will be useful for you. Haloalkanes and haloarenes we learn as a first chapter in our organic chemistry. So in this chapter, you will be coming across nearly 7 named reactions. Okay, so what are these 7 named reactions? So the first one I'll be taking here, I'll be giving you in such a way that you can relate one to another. Okay, that is the way I have designed it. Nimge use ಆಗ್ಬೋದು ಅಂತ ಅನ್ಕೋತೀನಿ. So the first one we have here Woods reaction or Woods reaction ಅಂತ ಕರಿತೀವಿ ನಾವು. So this is a well-known named reaction. In this case, we will be talking about haloalkanes. So you know haloalkanes, we will be representing them with a general formula that is Rx. Haloarenes bandaga, we will take them as ARX, where X is representing the halo group. Can be it is fluoro, chloro, bromo, iodo. Okay. So in case of Woods reaction, we will be taking alkyl halide that is Rx. So I told haloalkane irbodu ಅಥವಾ alkyl halide anta karibodu. So here the reaction is with the sodium metal, okay, and 2 molecules of your alkyl halide you will be considering. So the reaction takes place in presence of here dry ether. Okay, we need to carry out the reaction in dry ether condition. So these 2 halides from 2 molecules will be going away with sodium metal, I can say. So you can easily understand what is the byproduct of this reaction along with the main product. Look at it. R will combine with R here and the byproduct will be 2NaX. So R and R nothing but it is an alkyl group. So as I told, this is an alkyl halide we have here. So alkyl and alkyl group combines to give you another alkane that will be generally higher alkane. Okay, this is simply these reactions we call it as coupling reactions. 2 molecules will combine with sodium metal to give you higher alkane along with NaX. There is one more similar reaction in this chapter that is nothing but we call it as a fatigue reaction. Okay. So in case of fatigue reaction, ಏನಾಗುತ್ತೆ, instead of alkyl halide, we will be taking aryl halide. So you know the simple aryl halide is nothing but generally we take about benzene, right? So we will attach a halogen group to benzene, which I have written here it as aromatic aryl. Okay. So same reagent, that is we have reaction with the sodium metal and another aryl halide here, that is nothing but derivative of benzene, chlorobenzene or bromobenzene, anything you can take. Reaction here also takes place in presence of dry ether. Similar reaction, coupling reaction here. This NaX you can remove. So you can see in the reaction, the way I have written, there will be 2, these benzene rings will be fused like this and the byproduct will be 2NaX. This is nothing but we call it as 2 phenyl rings are there. It is called diphenyl or biphenyl. Okay, this is another reaction. Now let us take the third reaction, which is a combination of these Woods as well as fatigue here. So we call it as Woods fatigue reaction. Okay, Woods fatigue reaction. So you can easily tell me what is the reagent required here. Woods requires a Rx, right? And fatigue requires what? That is aryl halide. So we can take halogen attached to the benzene ring here. And here also the reaction will take place in dry ether condition. So let us fuse, that is coupling of these 2 reactants, and we get alkyl group attached to the benzene ring and byproduct will be 2NaX here. So this is nothing but alkyl benzene. Okay, so these three reactions are very much related to one another, easy to remember also. In all the cases, whether it is Woods fatigue is a combination of these two, so easy to remember. In all these, the reaction is simple coupling reactions. Two molecules of alkyl halide gives you alkane, two molecules of aryl halide gives you, that is diphenyl or biphenyl. Another alkyl with aryl halide gives you alkyl benzene. So it is easy to relate. And for all these reactions, we need reactive metal, that is sodium, because I can't change the metal. These are all renamed reactions, and condition required is dry ether. Okay, so these are the three important. Definitely you will find ilenagutandre this time as you have MCQs, they can give you this general equation and they will ask you to identify what is the name of reaction. Okay, or they may give you any one example and they may ask you to find out the product. So this is the logic of working this reaction. So hagagi maybe it is in the descriptive part or you can find it in the MCQ part. That's how this list of reactions what I have taken here. Fine. Yes. Now the other remaining four reactions still we have not yet over. These are like one set of reaction where you can relate to one another. Okay. Now let us take the next reaction, that is nothing but Finkelstein reaction. Later I'll give you few questions also simultaneously, you can work it out. Yes. Fourth one. So that is nothing but we have Finkelstein reaction. Yes. What is this reaction? This comes under your preparation part. Okay, preparation part of alkyl halide. Here also the reactant is Rx, but this X will be specifically in this case either chloro or bromo. Okay, previously I didn't give that condition. You can use any one of them, but there will be a difference in the rate of reaction. To that extent your questions won't be there, so you can directly look into the reaction. Here it is specific chloro or bromo, and this particular alkyl halide is made to react with what? Sodium iodide. Okay, this is very important. And at the same time, for this reaction, we need a dry acetone condition here. And reaction, look at it, easy to remember. You can remove again NaX and R and I will combine to give Ri. Okay, and the byproduct will be here NaX. That means we will be looking at what? We will be looking at iodoalkane preparation here. So this is a fixed reaction. Finkelstein reaction means it is mainly for the preparation of iodoalkane. Okay. So if you want to have a iodomethane bekuntidre, you have to start with the methyl chloride or methyl bromide. NaI is a fixed reagent. So as you are looking for iodo preparation here, you can remember the first after F you have a letter here I, so you can directly relate it to that. Sometimes we get confused which reaction is for what. Okay, so iodine iodoalkane preparation is from Finkelstein here. I later you can relate to that. Okay. The next reaction we have here, that is another Schwarz reaction. Okay, Schwarz reaction. Again, this reaction also comes under preparation of your alkyl halide only. Same, look at the reaction Rx. Here also X will be chloro or bromo, and then it is treated with any of the fluorinating agent. Generally we prefer silver fluoride. Okay, we can take other few fluorinating agents also, but generally preferred will be silver fluoride here. And reaction takes place here. You don't require any other condition, just the heating is sufficient. As silver has a tendency to react with the halide to get precipitated here, we can think of the reaction, that is Rf, and we have the byproduct will be AgX here. Okay, so you can see here this fluorine will be fixed, and this fluorine is coming from silver fluoride. This is called fluorinating agent. So we prepare here, that is fluoroalkane. So we get fluoroalkane from any other, that is chloro or bromoalkane. That reaction is called Schwarz reaction. Okay. Yes. Now these two reactions, previously I have mentioned those three reactions are coupling reactions. Okay. But these two reactions, you can see this X is substituted by iodine. Here the X is substituted by fluorine. So you can see both these reactions are here. They are nothing but which substitution? It is I minus, F minus, right? So it is nucleophilic substitution. So these two are nucleophilic substitution reaction. We call it as SN reaction. Both of them are nucleophilic substitution reactions. Okay. So both are very similar. These two are same. Only you are replacing here iodinating agent. Here it is fluorinating agent. Okay. Yes. Coming to the next set of reaction in this particular chapter, that is Sand-Meyer's reaction, haloarine preparation we have, right? Only one named reaction under haloarine preparation, that is Sand-Meyer's. Let us see what is this Sand-Meyer's here. And you know, these are all names, whatever we have, they are all the names of the scientist who has given that particular reaction. And you can just go through if you are interested, you know, history no nodebodu, how they exactly got it. Sometimes there will be some interesting stories behind how exactly they got through this particular reaction. Okay. For us now it is everything ready in the form of book. So we will be just reading and using it for our examination purpose. But if you are interested in knowing the history behind it, it will be quite interesting. No doubt about that. So we have a Sand-Meyer's reaction here. Okay. Meyer's reaction. What is this Sand-Meyer's reaction? We will start with the aniline. Hope by this level, all of you know what is the meaning of aniline. Amino substituted benzene is called aniline. And first step of this reaction will be a diazotization step. And students, this particular reaction you will again study under amines chapter. Okay. In amines chapter, under the reaction of aniline, this is the one important reaction you are studying. So there your burden will be reduced. ಈಗ ಗೊತ್ತಾಯ್ತು ಇಲ್ಲಿ ಬರುತ್ತೆ ಮತ್ತೆ ಅದೇ reaction ನೀವು amines chapter ಅಲ್ಲಿ ಓದ್ತೀರಾ. So ಹಾಗಾಗಿ you can relate and study. So we require here sodium nitrite, along with generally we go for hydrochloric acid, and this reaction has to be conducted under cold condition. Okay, 0 to 5 degree, because without that the product won't be formed. This amine group, NH2 group, gets converted into N2 plus Cl minus. This is called benzene diazonium chloride. Okay, so this is called benzene diazonium chloride. But this is not ultimate. This benzene diazonium chloride, we call it as an intermediate. This won't be stable enough. ಈಗ ನೀವು ಲ್ಯಾಬ್ ಅಲ್ಲಿ ಮಾಡಬೇಕಾದ್ರೂ ಈ ರಿಯಾಕ್ಷನ್ ಕ್ಯಾರಿ ಔಟ್ ಮಾಡಿದ ಕೂಡಲೇ ಈ ಟೆಂಪರೇಚರ್ ಇದ್ರೆ ಇದು ನಿಮಗೆ ಬರುತ್ತೆ. ಸೊ the moment you disturb the temperature, it cannot be stable. It will decompose to give you different products. So this is called intermediate. Further, you can drive the reaction based on your wish. Now, if I take here Sand-Meyer's reaction I am having, that's why I'll take the condition for Sand-Meyer's reaction is cuprous salt we will take along with the hydrogen halide, HX barita idini. So whichever the HX X you take, that will be substituted to the benzene ring. So you will get here X and the byproduct will be N2, liberation of nitrogen gas. So if I'm interested in preparing chlorobenzene here, you have to take HCl. If you are interested in preparing bromobenzene, you have to go for HBr. Okay. So that's how this reaction will be driven. The name of the reaction is Sand-Meyer's, which is for the preparation of haloarine. Okay. Preparation of haloarine. So these are important 6 reactions which is under your haloalkanes and haloarine. For sure, out of these, you will definitely come to come across 2 or 3, no doubt, either in 1 marks or in 2 marks or in the 5 marks question. So you know this blueprint. Just check what marks weightage you have for this chapter. Here and there, definitely you will come across this reaction. Maybe the one choice is giving you option, another is to name the reaction or to identify the product. All this, or they may ask for this conversion, what is the reagent required. So ಇದೆಲ್ಲ ಅದರಲ್ಲಿ ಬರುವಂತ ಪಾಸಿಬಲ್ ಕ್ವೆಶ್ಚನ್ಸ್. ಅದಕ್ಕೆ ಫಸ್ಟ್ ಯು ಶುಡ್ ನೋ ರಿಯಾಕ್ಷನ್ ಅಲ್ಲಿ ಏನಿದೆ ಅಂತ. ಈಗ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ನೇಮ್ಡ್ ರಿಯಾಕ್ಷನ್ ಅಂದಾಗ ಓಕೆ, ನೇಮ್ಡ್ ರಿಯಾಕ್ಷನ್ ಈಸಿ, ಇಂಟ್ರೆಸ್ಟಿಂಗ್ ಆಗಿರುತ್ತೆ. ಬಟ್ ವಿ ಶುಡ್ ರಿಮೆಂಬರ್ ಅಲ್ವಾ, ಇಲ್ಲಿ ಸಮ್ ಟೈಮ್ಸ್ ಯುವರ್ ಮೆಮೊರಿ ಇಸ್ ಡೆಫಿನೆಟ್ಲಿ ರಿಕ್ವೈರ್ಡ್. ನೀವು ಅದಕ್ಕೆ ಹೊಸದಾಗಿ ಏನೋ ಲಾಜಿಕ್ ಅಪ್ಲೈ ಮಾಡಕ್ ಆಗಲ್ಲ. ಈಗ ನನಗೆ ಸ್ವಾಟ್ಸ್ ರಿಯಾಕ್ಷನ್ ಅಂದ್ರೆ ಗೊತ್ತಿರಬೇಕು ಏನಾಗ್ತಾ ಇದೆ ಸ್ವಾಟ್ಸ್ ರಿಯಾಕ್ಷನ್ ಅಂತ, ಅದನ್ನೇ ಯಾರು ನಿಮ್ಗೆ ಹೇಳಕ್ ಆಗಲ್ಲ. ಸೊ ಓವರ್ ಎ ಪೀರಿಯಡ್ ಏನಾಗುತ್ತೆ, ನೀವು ಪ್ರತಿ ಸರ್ತಿ ವರ್ಕ್ ಮಾಡಿದಾಗ ನಿಮಗೆ ಫಿಕ್ಸ್ ಆಗುತ್ತೆ. ಆಲ್ಕೈಲ್ ಹ್ಯಾಲೈಡ್ ಜೊತೆ ಸಿಲ್ವರ್ ಫ್ಲೂರೈಡ್ ಬಂತು ಅಂದಾಗ ಹ್ಯಾಲೋಜನ್ ರಿಪ್ಲೇಸ್ ಆಗಿ ಫ್ಲೂರಿನ್ ಬರ್ತಾ ಇದೆ ಇಲ್ಲಿ, ಕ್ಲೋರಿನ್ ಇಂದ ಕ್ಲೋರಿನ್ ಬ್ರೋಮಿನ್ ರಿಪ್ಲೇಸ್ ಆಗಿ. ಸೊ ಅಷ್ಟು ಸ್ಕೆಲೆಟಲ್ ಈಕ್ವೇಶನ್ ಗೊತ್ತಿದ್ದಾಗ ಮಾತ್ರ ಯು ಕ್ಯಾನ್ ಅಪ್ಲೈ ಟು ದಟ್. ಓಕೆ, ಕ್ವಶನ್ ನೋಡಿದಾಗ ನಿಮ್ಗೆ ನೆನಪಾಗುತ್ತೆ. ಸೊ ಆ ಇನಿಷಿಯಲ್ ಸ್ಟೆಪ್ ಯು ಶುಡ್ ಡೂ, ಯು ಶುಡ್ ಪ್ರಾಕ್ಟೀಸ್ ಇಟ್, ಯು ಶುಡ್ ಲಿಟ್ ಔಟ್ ಅಂಡ್ ಜಸ್ಟ್ ಅಬ್ಸರ್ವಿಂಗ್ ಇಟ್. It's like every day if you have this list in front of you, just observe 1 or 2 times every day, it will be registered in your mind. That is how easily we can do it. Same as in maths formula, a plus b whole square andre enu anta when you started studying in your school, you have by hearted it so many times, right? Your maths teacher used to ask you 100 times the same formula. Ban kudle nimi formula ಹೇಳ್ಸ್ಬಿಡೋರು, a plus b whole square andre ಏನು ಅಂತ. You used to tell a square plus b square ಅಂತ ಹೇಳ್ಬಿಟ್ಟು ಸ್ಟಾರ್ಟ್ ಮಾಡ್ತಾ ಇದ್ರಿ. ಸೇಮ್ ಆರ್ಗ್ಯಾನಿಕ್ ಕೆಮಿಸ್ಟ್ರಿ ನಲ್ಲೂ ನೇಮ್ಡ್ ರಿಯಾಕ್ಷನ್ಸ್ ಯು ಹ್ಯಾವ್ ಟು ರಿಪೀಟ್ ಮೆನಿ ಟೈಮ್ಸ್. ಇದಂದ್ರೆ ಇದು ಅಂತ ನನಗೆ ಗೊತ್ತಿರ್ಬೇಕು, ಆಗ್ಲೇ ನಾನು ಮಾಡಕ್ಕಾಗೋದು. ಸೊ ದಟ್ಸ್ ಹೌ ದಿಸ್ ಲಿಸ್ಟ್ ಯು ಶುಡ್ ಅಬ್ಸರ್ವ್. ಓಕೆ, ಎಸ್. ಸೊ ಇದು ಮೇನ್ ರಿಯಾಕ್ಷನ್. ಇದು ಬಿಟ್ರೆ ನಿಮಗೆ ಹ್ಯಾಲೋ ಅರೀನ್ಸ್ ಅಲ್ಲಿ ಸಬ್ಸ್ಟಿಟ್ಯೂಷನ್ ರಿಯಾಕ್ಷನ್ಸ್ ಬರ್ತವೆ. ಆ ಸಬ್ಸ್ಟಿಟ್ಯೂಷನ್ ರಿಯಾಕ್ಷನ್ಸ್ ನ ನಾವು ಆರೋಮ್ಯಾಟಿಕ್ ಹೈಡ್ರೋಕಾರ್ಬನ್ಸ್ ಅಲ್ಲಿ ಒಂದ್ಸತಿ ಡಿಸ್ಕಸ್ ಮಾಡಿದ್ವಿ. ಅಂದ್ರೆ ಅದು ಸಿಮಿಲರ್ ರಿಯಾಕ್ಷನ್ ಬರುತ್ತೆ, ಫ್ರಿಡಲ್ ಕ್ರಾಫ್ಟ್ಸ್ ಆಲ್ಕೈಲೇಷನ್ ಅಂಡ್ ಅಸೈಲೇಷನ್. ದಟ್ ಈಸ್ ಅನದರ್ 2 named reactions in this chapter. So I'll just quickly give you those reactions, and those 2 reactions generally they are like in other chapters also you will come across. Okay, but some of the cases may be based on the functional group, you may not have those reactions also. Otherwise they are the like general reaction I can say. Okay, one is Friedel Crafts alkylation. Okay, Friedel Crafts alkylation. Another one is acylation. Same name, alkylation, or I can say it is acylation reaction here. Fine, acylation reaction here. Fine, any messages or something? I don't think. Okay, fine. Acylation. So in this case, if I consider here any chlorobenzene, I'll take chlorobenzene. You can take any bromobenzene. ಯಾವುದು ತಗೋಬಹುದು ಇಲ್ಲಿ. Okay, ಮತ್ತೆ reactivity ನಿಮಗೆ ಡಿಫರೆನ್ಸ್ ಆಗೋ ಚಾನ್ಸಸ್ ಇದೆ. Okay, fine. Now if I take this chlorobenzene, ಎರಡು ರಿಯಾಕ್ಷನ್ ಜೊತೆಲಿ ಬರೀತೀನಿ. ಸೊ ಯು ಕ್ಯಾನ್ ಜಸ್ಟ್ ಅಬ್ಸರ್ವ್ there is a difference with respect to reagent taken. When I say alkylation, ಎಲ್ಲರಿಗೂ ಗೊತ್ತು alkyl group ಅಂದ್ರೆ ಏನು. Any alkyl, it can be methyl, ethyl. Okay, simple. Let me take here methyl. Any methyl halide we should take. So methyl chloride ತಗೋತೀನಿ. Another reagent is anhydrous aluminum chloride. ಇದು ಗೊತ್ತಿರಬೇಕು. So when you see this combination, any RX with anhydrous AlCl3, if you take any aromatic ring system like maybe simple benzene, or you can take chlorobenzene, bromobenzene or nitrobenzene, ಆಗೆಲ್ಲ ನಿಮಗೆ ಈ ರಿಯಾಕ್ಷನ್ ನ ನೆನಪು ಮಾಡ್ಕೋಬೇಕು. Friedel Crafts alkylation ಏನಾಗ್ತದೆ alkylation ಅಲ್ಲಿ, we can see this alkyl part, that's why I call it as alkylation. Okay, sometimes we use a term alkylation ಅಥವಾ halogenation ಅಂತೀವಿ. ಅಂದ್ರೆ halogen ಆಡ್ ಆಗುತ್ತೆ ಅಥವಾ halogen substitute ಆಗುತ್ತೆ. Then we say hydrogenation, addition of hydrogen ಅಂತ ಹೇಳ್ತೀವಿ. ಅದೇ ತರ alkylation ಅಂದ್ರೆ for the given substrate you are carrying out alkylation. So where this alkyl group will go, you have studied about this chloro group. Okay, it is electron donating group, so it is a ortho para director. So we'll be ending up in having 2 products. One is we get here chloro, in the para position the alkyl group will be. Another we can get here alkyl group will be substituted in the ortho position. This is called para-chlorotoluene. You can call it as, this is ortho product. What is the difference in the next reaction? Here I need to go for acyl group. Understand, acyl group. So we have to take acetyl chloride, CH3COCl. Another is AlCl3 only, anhydrous. Okay, yes, because this acts as a Lewis acid. ನಿಮಗೆ ಗೊತ್ತಿರುತ್ತೆ, Lewis acid will generate the electrophile. Here the electrophile is this acyl group. Okay, so acyl group gets substituted ortho as well as in the para. Generally in this case, you know, the para will be the major product and ortho will be the minor product. Okay, so these are the para-chloroacetophenone. This is a ketone group, acetophenone, or orthochloro like acetophenone you can get as a product. Okay, generally we write the major product first and minor product next. This is Friedel Crafts acylation reaction here. So what do you observe in this case? ಏನು nature of reaction if I ask? I can see here this is plus, This is also plus. Nothing but they are the electrophiles. You know the notation of electrophile. And this electrophile is substituted to what? Benzene ring. Either we have substituted hydrogen in the ortho position or we have substituted hydrogen in the para position, right? So what do you call it as? This is called substitution, like electrophilic substitution to which system? To the aromatic ring, because we are substituting to the aromatic ring, hence it is called electrophilic substitution aromatic. Okay, so that is a mechanism which you have learned in first year. Fine. So these are the 7 important named reactions in this particular chapter. Okay, maybe how the questions will be asked, you told. Now you have observed few things, right? So if I write few reactions on the board, you could be able to identify the name as well as product. Okay, that's how you can learn the organic chemistry. Now I'll give you just 1 or 2 sample questions because we have a limitation of time, right? So in case if I write some working examples, I'll take here. Fine. Yes. Observe the reaction. Let me consider here some methyl bromide. Okay. And let me write over the arrow mark. The first condition is sodium in presence of, or I can write sodium in presence of dry ether. The moment ನೀವು sodium dry ether with RX ನೋಡಿದ ಕೂಡಲೇ you should recall this is a Wurtz reaction, right? Now remove this Br, attach it to the sodium here, club this methyl group. So CH3 will become what? It will become C2H6. This is how you decide the product. Okay, na? ಇನ್ನೊಂದು reaction ಬರಿತೀನಿ ಇಲ್ಲಿ. So for example, I'll take aniline. Okay, the moment I see aniline, and I'll give you the condition over the arrow mark. First condition in baritini NaNO2 with HCl at low temperature 0 to 5 degree anta baritini. Second condition I'll write Cu2 Br2 with HBr baritini illi. So in the previous case I have given you some general reaction. ಈಗ ನಾನು specific ಆಗಿ ಬರಿತೀನಿ. So ಇಲ್ಲಿ ಅನಿಲಿನ್ ನೋಡಿ ಡಯಾಜೋಟೈಸೇಷನ್ ನೋಡಿದ ಕೂಡಲೇ ನಿಮ್ಗೆ ಅರ್ಥ ಆಗಬೇಕು ಇದು ಸ್ಯಾಂಡ್ ಮೇಯರ್ಸ್ ಅಲಾಂಗ್ ವಿತ್ ಈ ಕಂಡೀಷನ್. ಓಕೆ, ಕ್ಯುಪ್ರಸ್ ಹಲೈಡ್ ವಿತ್ HBr ಕೊಟ್ಟಿದ್ದೀನಿ. ಸೊ Br ಇರೋದ್ರಿಂದ ಇಲ್ಲಿ NH2 ಗ್ರೂಪ್ ನಮ್ಗೆ ಏನಾಗುತ್ತೆ, ಇಟ್ ಗೆಟ್ಸ್ ಕನ್ವರ್ಟೆಡ್ ಇನ್ ಟು ಬೆನ್ಸಿನ್ ಡಯಾಸೋನಿಯಂ ಕ್ಲೋರೈಡ್ ಅಂಡ್ ನೌ ಇನ್ ದ ಸೇಮ್ ಪೊಸಿಷನ್ ಐ ಕ್ಯಾನ್ ಫೈಂಡ್ ದ Br. ದಿಸ್ ಇಸ್ ಅ ಪ್ರಿಪರೇಷನ್ ಆಫ್ bromobenzene. So what is the reaction? It is nothing but ಸ್ಯಾಂಡ್ ಮೇಯರ್ಸ್ ರಿಯಾಕ್ಷನ್, ರೈಟ್? ಸ್ಯಾಂಡ್ ಮೇಯರ್ ರಿಯಾಕ್ಷನ್. ಹೀಗೆ ಐಡೆಂಟಿಫೈ ಮಾಡೋದು. Or I can write another example for you here. Okay, let me take methyl chloride. Okay, I may write over the arrow mark I have sodium iodide and a dry acetone condition. Okay, look at it. ಈಗ ಇದಕ್ಕೆ ಒಂದೊಂದ್ಸತಿ ರಿಯಾಕ್ಷನ್ ಈಸಿಯಾಗಿ ಐಡೆಂಟಿಫೈ ಮಾಡ್ಬಿಡ್ಬೋದು. ಏನಿದೆ Na+ and I-. ಈಗ ಎರಡರ ಮಧ್ಯೆ ರಿಯಾಕ್ಷನ್ ಆಗ್ತಿದೆ ಅಂದ್ರೆ ಓನ್ಲಿ ಪಾಸಿಬಿಲಿಟಿ ಏನು, I- will come and attack this, displace this, right? So there will be a formation of methyl iodide and NaX. So which is this reaction? Iodoalkane you are preparing. And ಹೇಳ್ದೆ ನಾನು iodoalkane ಅಂದ್ರೆ you have to remember it is a Finkelstein reaction. Okay, so this is how you should see, observe the substrate and reactant. A combination ಗೊತ್ತಿರಬೇಕು named reaction ಗೆ. Then you can easily identify the reaction and you can also easily identify the product. Okay, just few sample examples I have added here. So this is your entire the first chapter, that is haloalkanes and haloarenes. ಈಗ we will look into the next chapter. ಏನಿದೆ next chapter? Alcohols and phenols along with ethers. In that chapter actually ನಿಮಗೆ ತುಂಬಾ ಲೆಸ್ ನಂಬರ್ ಆಫ್ ನೇಮ್ಡ್ ರಿಯಾಕ್ಷನ್ಸ್ ಇರೋದು, ಬಟ್ ಇರೋದಷ್ಟು ಇಂಪಾರ್ಟೆಂಟ್. ಎಲ್ಲೋ ಒಂದ್ ಕಡೆ ಕೇಳ್ತಾರೆ ಆ ಎಲ್ಲಾ ರಿಯಾಕ್ಷನ್ಸ್. ಹಾರ್ಡ್ಲಿ ಯು ಕ್ಯಾನ್ ಕೌಂಟ್. ದರ್ ವಿಲ್ ಬಿ ಓನ್ಲಿ 4 ರಿಯಾಕ್ಷನ್ಸ್ ಇನ್ ದಟ್ ಕೇಸ್. ದಟ್ ಈಸ್ ಒನ್ ಇಸ್ ಕೋಲ್ಬೇಸ್ ರಿಯಾಕ್ಷನ್ ಬರುತ್ತೆ, ಇನ್ನೊಂದು ರಿಮರ್ಟಿಮನ್ ರಿಯಾಕ್ಷನ್ ಬರುತ್ತೆ, ನೆಕ್ಸ್ಟ್ ಯು ವಿಲ್ ಬಿ ಹ್ಯಾವಿಂಗ್ ಲೂಕಾಸ್ ಟೆಸ್ಟ್, ಇನ್ನೊಂದು ವಿಲಿಯಮ್ಸನ್ ಸಿಂಥೆಸಿಸ್. ಅಷ್ಟೇ ಬರೋದು. ಬೇರೆಲ್ಲ ಯು ಹ್ಯಾವ್ ಸಮ್ ಅದರ್ ವೇ ಲೈಕ್ ಹೈಡ್ರೋಬೋರೇಷನ್ ಆಕ್ಸಿಡೇಷನ್ ಅಂತದೆಲ್ಲ ಇದೆ, ಬಟ್ ನೇಮ್ ಆಫ್ ದ ಸೈಂಟಿಸ್ಟ್ ಇದ್ದು ಇರುವಂತ ರಿಯಾಕ್ಷನ್ಸ್ ನಿಮಗೆ 4 ಸಿಗೋದು ಆಲ್ಕೋಹಾಲ್ಸ್ ಫಿನಾಲ್ಸ್ ಅಲ್ಲಿ. ಸೊ ಆಲ್ಮೋಸ್ಟ್ 4 ನೀವು ಆಲ್ಮೋಸ್ಟ್ ಪೇಪರ್ ಅಲ್ಲಿ ನೋಡ್ತೀರಾ, ಒಂದಿಲ್ಲ ಒಂದು ಪಾರ್ಟ್ ಅಲ್ಲಿ ಬಂದೇ ಬರುತ್ತೆ. ಓಕೆ, ಸೊ ಲೆಟ್ ಅಸ್ ಸಿ ಆಲ್ ದೋಸ್ ರಿಯಾಕ್ಷನ್ಸ್ ನೌ. In that, the first one what I'm talking about, that is Lucas test. ನೀವು ಕೇಳಿರ್ತೀರಾ, ನಿಮ್ ಟೀಚರ್ಸ್ ಹೇಳಿರ್ತಾರೆ ಡಿಸ್ಕಸ್ ಮಾಡ್ಬೇಕಾರೆ. This is a test to distinguish. ಹಾಗಾಗಿ ಅದನ್ನ ಕೇಳ್ತಾರೆ. ಈವನ್ ನಿಮ್ಗೆ ಕಾಂಪಿಟೇಟಿವ್ ಎಕ್ಸಾಮ್ ಅಲ್ಲೂ ಅದನ್ನ ಇಂಡೈರೆಕ್ಟ್ ಆಗಿ ಕ್ವೆಶ್ಚನ್ ಮಾಡ್ತಾರೆ. So let us talk about here the next chapter, that is alcohols, phenols and ethers. Okay, ethers, less number of reactions. Hope we can quickly complete it. ಫಸ್ಟ್ ಒನ್ ಇಲ್ಲಿ ನೋಡಿ ಏನಿದೆ, ವಾಟ್ ಡಿಡ್ ಐ ಸೇ, ದಟ್ ಈಸ್ ಲೂಕಾಸ್ ಟೆಸ್ಟ್. ಟೆಸ್ಟ್ ಅಂತ ಬರೆದಿದ್ದೀನಿ. ವಾಟ್ ಇಸ್ ದಿಸ್ ಟೆಸ್ಟ್? ಇದು ಆಲ್ಕೋಹಾಲ್ಸ್ ಗೆ ಟೆಸ್ಟ್. ಪ್ರೈಮರಿ, ಸೆಕೆಂಡರಿ, ಟರ್ಷರಿ ಡಿಸ್ಟಿಂಗ್ವಿಶ್ ಮಾಡೋದು ಹೇಗೆ ಅನ್ನೋದನ್ನ ಈ ಟೆಸ್ಟ್ ಅಲ್ಲಿ ನಮ್ಗೆ ಗೊತ್ತಾಗುತ್ತೆ. ಸೊ ದಟ್ಸ್ ವೈ ವಿ ಕಾಲ್ ಇಟ್ ಅಸ್ ಲೂಕಾಸ್ ಟೆಸ್ಟ್. ಸೊ ಯೂಶಲಿ 1 ಮಾರ್ಕ್ಸ್ ಕ್ವೆಶ್ಚನ್ ನಿಮಗೆ ಕೇಳ್ತಾರೆ ಲೂಕಾಸ್ ರಿಯೇಜೆಂಟ್ ಏನು ಅಂತ. ಸೊ ವಾಟ್ ಇಸ್ ದಿಸ್ ಲೂಕಾಸ್ ರಿಯೇಜೆಂಟ್? ಇಟ್ ಇಸ್ ನಥಿಂಗ್ ಬಟ್ ಕಾನ್ಸಂಟ್ರೇಟೆಡ್ ಹೈಡ್ರೋಕ್ಲೋರಿಕ್ ಆಸಿಡ್ ಜೊತೆ ಜಿಂಕ್ ಕ್ಲೋರೈಡ್ ಇರುತ್ತೆ. ಓಕೆ, ಅನ್ಹೈಡ್ರಸ್ ಜಿಂಕ್ ಕ್ಲೋರೈಡ್ ಇರುತ್ತೆ. ದಟ್ ಇಸ್ ಅ ಲೂಕಾಸ್ ರಿಯೇಜೆಂಟ್ ಯು ಹ್ಯಾವ್. So how the reaction here? Reaction, general reaction pretty simple. ಏನದು? ROH, alcohol ತಗೋತೀನಿ ನಾನಿಲ್ಲಿ. Okay, ROH. With what is the reaction here? Reaction is with the HCl, that is concentrated. Oh, the arrow mark we have, that is anhydrous zinc chloride. Let me take. Okay, reaction is pretty simple. We can see OH is there, H is there, so it can go out as water and Cl will be substituted. So we have here formation of RCl. Reaction is simple. ಈಗ ಇಲ್ಲಿ story starts here. Whether you have the alcohol is primary, secondary or tertiary ಅನ್ನೋದನ್ನ ಇದರಿಂದ ಡಿಸೈಡ್ ಮಾಡ್ತೀವಿ. ಏನಾಗುತ್ತೆ? ಪ್ರೈಮರಿ ಆಲ್ಕೋಹಾಲ್, ಈಗ ನೀವು ಪ್ರೈಮರಿ ಆಲ್ಕೋಹಾಲ್ ಒಂದು ಎಕ್ಸಾಂಪಲ್, ಈಗ ನಾನು ಇಲ್ಲಿ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ CH3, CH2, CH2OH ತಗೊಳ್ತೀನಿ. Okay, so that it is easy for you to understand. Or any primary alcohol I can take, starting from methanol also I can take. I'll take ethanol. You know the meaning, right? This carbon is primary carbon, so it is called primary alcohol. Now if I take another example for secondary alcohol, that is a OH group should be attached to the secondary position. This is your secondary alcohol. Tertiary, let me take here RCCH3. Carbon, nature of the carbon should be tertiary. OH is attached to this, so this is a tertiary carbon. Okay, tertiary alcohol, OH group attached to tertiary carbon. This is secondary alcohol. For all this, I'll take in 3 beakers. One is ethanol, another one is propan-2-ol, another one is 2-methyl propan-2-ol. You can take 3 beakers ಅಲ್ಲಿ ಇಟ್ಕೊಳ್ಳಿ. ಎಲ್ಲದಕ್ಕೂ ಲೂಕಾಸ್ ರಿಯೇಜೆಂಟ್ ಹಾಕಿ. ಏನಾಗುತ್ತೆ ಆಗ? ಸೊ ಪ್ರೈಮರಿ ಯು ಕ್ಯಾನ್ ಸಿ ಇಮಿಡಿಯೇಟ್ ಟರ್ಬಿಡಿಟಿ ಬರುತ್ತೆ. ಅಂದ್ರೆ ದಿಸ್ ಪ್ರೈಮರಿ ವಿಲ್ ರಿಯಾಕ್ಟ್ ವಿತ್, ಈಗೆಲ್ಲದಕ್ಕೂ I'll make it like a common. So we'll be having here ಲೂಕಾಸ್ ರಿಯೇಜೆಂಟ್. ಈಗ ಲೂಕಾಸ್ ರಿಯೇಜೆಂಟ್ ತಗೊಂಡು ಟೆಸ್ಟ್ ಮಾಡಿದಾಗ, in case if it is primary, we will find the observation is immediate turbidity. Okay, because your alcohol is getting converted into, in this case, one minute, one mistake we have done. Okay, so alcohol in this case, what happens? They have the reactivity difference. Now what you are getting? Alkyl halide. Give me a minute. Alkyl halide. ಆಲ್ಕೋಹಾಲ್ ವಾಟರ್ ಸಾಲ್ಯುಬಲ್ ಆಗುತ್ತೆ. ಆಲ್ಕೈಲ್ ಹ್ಯಾಲೈಡ್ ಸಾಲ್ಯುಬಿಲಿಟಿ ಕಡಿಮೆ ಇರುತ್ತೆ. ಸೊ ಹಾಗಾಗಿ ಏನಾಗುತ್ತೆ, ಆಲ್ಕೈಲ್ ಹ್ಯಾಲೈಡ್ ಫಾರ್ಮ್ ಆದ ಕೂಡಲೇ ಯು ವಿಲ್ ಬಿ ಗೆಟಿಂಗ್ ದ like turbidity there. So there will be a like immediate turbidity you can observe in that case. So based on the alcohol, if I take primary, okay, so you need to understand why turbidity ಆಗಿ ಬರ್ತಿದೆ ಅಂತ ಹೇಳಿದ್ರೆ, ಹ್ಯಾಲೈಡ್ ಬಂದ ಕೂಡಲೇ ಸಾಲ್ಯುಬಿಲಿಟಿ ಕಡಿಮೆ ಆಗೋದ್ರಿಂದ ಟರ್ಬಿಡಿಟಿ ಅಪಿಯರ್ ಆಗುತ್ತೆ. ಸೊ ಇನ್ ದಿಸ್ ಕೇಸ್ ಇಫ್ ಯು ಟೇಕ್ ಟರ್ಷರಿ, ಟರ್ಬಿಡಿಟಿ ನಿಮಗೆ ಇಮಿಡಿಯೇಟ್ ಆಗಿ ಫಾರ್ಮ್ ಆಗುತ್ತೆ ಇನ್ ಕೇಸ್ ಆಫ್ ಟರ್ಷರಿ ಆದ್ರೆ. ಸೆಕೆಂಡರಿ ಆದ್ರೆ ಆಫ್ಟರ್ 5 minutes. ಪ್ರೈಮರಿ ಆದ್ರೆ ಹಾಗೆ ಬಿಟ್ರೆ ನಿಮ್ಗೆ ಫಾರ್ಮೇ ಆಗಲ್ಲ. ಓಕೆ, ಇಫ್ ಯು ಕೀಪ್ ಇಟ್ ಲೈಕ್ ದಟ್ ಓನ್ಲಿ, there won't be like turbidity formed easily. So I can say, so if there is no turbidity at room temperature, ಯಾಕಂದ್ರೆ ಹಾಗೆ ಬಿಟ್ರೆ ಅಂತ ಅಂದೆ, ಹಾಗೆ ಬಿಟ್ರೆ ಅಂದ್ರೆ ರೂಮ್ ಟೆಂಪರೇಚರ್, ಹೀಟ್ ಮಾಡಿದ್ರೆ there could be a possibility. ಹಾಗಾಗಿ ಕಂಡೀಷನ್ಸ್ ಆರ್ ಇಂಪಾರ್ಟೆಂಟ್. ಸೇಮ್ ಕ್ವೆಶ್ಚನ್ ನಿಮಗೆ ಲಾಸ್ಟ್ ಇಯರ್ ಕಾಂಪಿಟೇಟಿವ್ ಎಕ್ಸಾಮ್ ನೀಟ್ ಅಲ್ಲಿ ಕೇಳಿದ್ರು, ಹೇಗೆ ಫಾರ್ಮ್ ಆಗುತ್ತೆ ಅನ್ನೋದನ್ನ ಸ್ಟೇಟ್ಮೆಂಟ್ ಕೈಂಡ್ ಆಫ್ ಕ್ವೆಶ್ಚನ್ ಕೇಳಿದ್ರು. ಸೊ ಇನ್ ಕೇಸ್ ಇಫ್ ಇಟ್ ಇಸ್ ಅ ಟರ್ಷರಿ, ಐ ಕ್ಯಾನ್ ಫೈಂಡ್ it is forming immediate turbidity here. Okay, immediate turbidity ಅಂದ್ರೆ solution will change into a turbid. Clear solution ಇರೋದು turbid ಆಗಿ change ಆಗುತ್ತೆ. So in case of secondary, the turbidity appears, so we can say after 5 minutes. Okay, appears slow ಆಗಿ, reaction appears after some time, after 5 minutes generally we refer. Okay, so here no turbidity, but no turbidity at what temperature? Room temperature. That's important. Okay, so that's how I can distinguish. So if you have alcohol from any source, on the alcohol ತಗೊಳ್ಳಿ ಎಲ್ಲಿಂದಾರು, just add the Lucas reagent. If you get a turbidity, you can conclude it as tertiary. If there is no turbidity at all, you can conclude it as primary. If turbidity appears ಸ್ವಲ್ಪ ಹೊತ್ತು ಅಲ್ಲೇ ಬಿಟ್ಟು ನೀವು ರಿಯಾಕ್ಷನ್ ಟೆಸ್ಟ್ ಟ್ಯೂಬ್ ಅಲ್ಲೇ ಬಿಟ್ಟು ಆಮೇಲೆ ಕಾಣಿಸಿದ್ರೆ, you can conclude it as secondary alcohol. ಇದು ಒಂದು ಇಂಪಾರ್ಟೆಂಟ್ ರಿಯಾಕ್ಷನ್. 1 marks ರಿಯೇಜೆಂಟ್ ಆರು ಕೇಳ್ಬೋದು, ಅಥವಾ how these 3 alcohols are distinguished with the help of Lucas reagent ಅನ್ನೋದನ್ನ ಖಂಡಿತ ಕೇಳ್ತಾರೆ. ಒಂದಿಲ್ಲ ಒಂದ್ ಪಾರ್ಟ್ ಅಲ್ಲಿ ಇದ್ದೇ ಇರುತ್ತೆ. ಓಕೆ, ಸೊ ಬಟ್ ಅದ್ರ ಜೊತೆಗೆ ಏನಾಗುತ್ತೆ, ಒಂದೊಂದ್ಸತಿ ನಮ್ಗೆ ಇದೆಲ್ಲ ಗೊತ್ತಿರುತ್ತೆ. ಐ ನೋ ಟರ್ಷರಿ ಇಮಿಡಿಯೇಟ್ ಟರ್ಬಿಡಿಟಿ ಫಾರ್ಮ್ ಆಗುತ್ತೆ ಅಂತ ಎಲ್ಲ ಗೊತ್ತಿರುತ್ತೆ. ಬಟ್ ಆ ಒಂದು ಕೇಸಲ್ಲಿ ಸಮಟೈಮ್ಸ್ ವಿ ಫೈಂಡ್ ಡಿಫಿಕಲ್ಟ್ ಇನ್ ಲೈಕ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡಿಂಗ್ ಪ್ರೈಮರಿ ಸೆಕೆಂಡರಿ ಟರ್ಷರಿ ಅಂದ್ರೆ ಏನು ಅಂತ ಐಡೆಂಟಿಫೈ ಮಾಡಕ್ ಆಗಿಲ್ಲ ಅನ್ನೋ ಕಂಡೀಷನ್ ಬಂದಾಗ ನಿಮಗೆ ಇಷ್ಟು ಟೆಸ್ಟ್ ಡೀಟೇಲ್ ಗೊತ್ತಿದ್ರೂ ಯೂಸ್ ಆಗಲ್ಲ. ಓಕೆ, ಸೊ ವಿ ಕ್ಯಾನ್ ಅದು ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್ ಆಗುತ್ತೆ. ಟರ್ಷರಿ ಅಂದ್ರೆ ಯಾವುದು, ಸೊ ದಟ್ಸ್ ವೈ ನೇಚರ್ ಆಫ್ ಕಾರ್ಬನ್ ನಿಮಗೆ ಕರೆಕ್ಟ್ ಆಗಿ ಗೊತ್ತಿರಬೇಕು. ಇಲ್ಲಿ ಪ್ರೈಮರಿ ಕಾರ್ಬನ್ ಇನ್ನೊಂದು ಕಾರ್ಬನ್ಗೆ ಅಟ್ಯಾಚ್ ಆಗಿದೆ. ದಿಸ್ ಈಸ್ ಸೆಕೆಂಡರಿ ಕಾರ್ಬನ್, ದಿಸ್ ಈಸ್ ಟರ್ಷರಿ ಕಾರ್ಬನ್. ಸೊ ಆ OH ಗ್ರೂಪ್ ಟರ್ಷರಿ ಕಾರ್ಬನ್ಗೆ ಅಟ್ಯಾಚ್ ಆಗಿದ್ರೆ ಟರ್ಷರಿ, ಸೆಕೆಂಡರಿಗೆ ಅಟ್ಯಾಚ್ ಆಗಿದ್ರೆ ಸೆಕೆಂಡರಿ, ಪ್ರೈಮರಿಗಾದ್ರೆ ಪ್ರೈಮರಿ. ಸೊ ಅದು ಗೊತ್ತಿಲ್ಲ ಅಂದ್ರೆ ನೀವು MCQs ಮಾಡಕ್ ಕಷ್ಟ ಆಗುತ್ತೆ. ಎಲ್ಲ ಗೊತ್ತಿದ್ದು ಇದು ಡಿಫಿಕಲ್ಟಿ ಆದ್ರೆ MCQ ಸಾಲ್ವ್ ಮಾಡಕ್ ಆಗಲ್ಲ. ಸೊ ಹ್ಯಾವ್ ಟು ಹ್ಯಾವ್ ಎ clear idea about what is primary, secondary, tertiary. Okay, then you can use it to identify the like answers. Fine. Yes, one reaction idu. Next one. Next two reactions are from phenols. Idu one alcohol allirodu, innond eradu barodu phenol alli. One is coal-base reaction. Another one is Riemann-Tiemann reaction. ಎರಡು ತುಂಬಾ close reactions. Hardly there is a difference, I can say. Both are reactions of phenol. So first we need to take phenol. ಎಲ್ಲರಿಗೂ ಗೊತ್ತು hydroxybenzene is phenol. So after this, coal-base reaction alli, the one of the reactant what we are taking is carbon dioxide. Carbon dioxide basically ನೋಡಿದ್ರೆ ಎಂತ ಅಂಥ reactive reagent ಅಲ್ಲ. Okay, so what happens, as I'm talking about reaction of carbon dioxide as a electrophile, first ನೀವು phenol ನ phenoxide ಆಗಿ convert ಮಾಡ್ಬೇಕಿಲ್ಲಿ here. So the reaction, coal-base reaction is nothing but reaction of sodium phenoxide with carbon dioxide at a bit higher temperature and pressure gives you what, an intermediate, which further on acidification gives you salicylic acid. So a story, narrating story you should know. Okay, that's the first sentence you need to write in your board exam. Illi, so first we get sodium phenoxide, the base you have, this is acidic, so you know that story. Okay, so hope board is visible, right? Sodium phenoxide you will get. Further you can make it to react with carbon dioxide. Second condition is acidification, so you can go for a HCl here. Okay, acidification, HCl. ಈಗ ಇಲ್ಲಿ ನೋಡಿ ಏನು ಈಸಿ, ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ನೆನಪಿದ್ರೆ this carbon dioxide in the ortho position we can find getting substituted as carboxylic group COOH, and this ONA as I'm adding HCl gets converted into OH. So look at the entire reaction. What do you find? I have replaced hydrogen from the benzene ring, ortho positional replace ಮಾಡಿ I have placed a carboxylic group. So ortho substituted this carboxylic group, we call it as a common name I am writing, that is salicylic acid. Athwa if you want to go for IUPAC nomenclature, this is the major main principal functional group. This is second position, so it will be 2-hydroxy, right? Benzene carboxylic acid. Okay, benzene carboxylic acid. Yes, so this is like usually for the 2 marker they will ask either in the part of like you can say 2 marks, or you can find sometimes even in the case of like I can say 1 mark also identifying the reaction. Second one, as I told, people will call it as Riemann-Tiemann or Riemann-Tiemann reaction. So we can look at this reaction. Yes, Riemann-Tiemann reaction. Fine. What is the reaction here? The same phenol. I told both are the reactions of phenol. So together you can learn this. Definitely the first condition we will be going here also with sodium hydroxide, because sodium phenoxide will be like we can find easily like more reactive. Along with the sodium hydroxide we also take another reagent, that is chloroform. Idu nenpirbeku, chloroform ide annodu gottirbeku, adne heltirodu. Initially you have to put an effort to understand the reaction. So when you treat these two with the phenol, we get first step an intermediate, that is CHCl2, kind of we can find benzylidene intermediate we will get. This intermediate further reaction with NaOH, that is aqueous. Step wise ಬರಿತಾ ಇದೀವಿ ಯಾಕಂದ್ರೆ ಈ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಮೇಲೇನು ಯು ಹ್ಯಾಡ್ ಎ ಕಾಂಪಿಟೇಟಿವ್ ಎಕ್ಸಾಮ್ ಕ್ವೆಶ್ಚನ್. ಒಂದೊಂದ್ಸತಿ ಡೈರೆಕ್ಟ್ ಪ್ರಾಡಕ್ಟ್ ಕೇಳ್ತಾರೆ, ಒಂದೊಂದ್ಸತಿ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಏನ್ ಫಾರ್ಮ್ ಆಗಿದೆ ಅಂತ ಕೇಳ್ತಾರೆ. ಸೊ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಐಡಿಯಾ ಇರಬೇಕು. ಸೊ NaOH ಜೊತೆ ಟ್ರೀಟ್ ಮಾಡಿದಾಗ ಏನೇನೋ ಆಗುತ್ತೆ. ದಿಸ್ NaOH ವಿಲ್ ಹೆಲ್ಪ್ ಯು ಟು ಜನರೇಟ್ ದ ಇಲೆಕ್ಟ್ರೋಫೈಲ್. ಇಲ್ಲಿ CCl2 ಡೈಕ್ಲೋರೋಕಾರ್ಬಿನ್ ಅನ್ನೋ ಇಲೆಕ್ಟ್ರೋಫೈಲ್ ಜನರೇಟ್ ಆಗುತ್ತೆ. ಇಲೆಕ್ಟ್ರೋಫೈಲ್ ಇಲ್ಲಿ ಸಬ್ಸ್ಟಿಟ್ಯೂಟ್ ಆಗುತ್ತೆ. CCl2 ಅಂದ್ರೆ ಹೈಡ್ರೋಜನ್ ಇಲ್ಲಿರುತ್ತೆ, ಸೊ ಇಟ್ ವಿಲ್ ಫಾರ್ಮ್ CHCl2. With aqueous NaOH we will get the product along with further one step I can write here only, that is acidification. Again you have to go for, so OH and we can find here CHO. Okay, now look at the reaction. ಎರಡು ಕಡೆ ನಿಮಗೆ ಒಂದು ಸ್ವಲ್ಪ ಸಿಮಿಲಾರಿಟಿ ಕಾಣಿಸುತ್ತೆ. ಏನದು, ಇಲ್ಲಿ ನಾನು 1, 2 ಅಂತ ಬರ್ದಿದ್ದೀನಿ, ನೀವು ಇನ್ನೊಂದು ಸ್ಟೆಪ್ ಬರ್ದು ಬರಿಬಹುದು. ಓಕೆ, ಸೊ acidification I have brought here because space problem. Now look at the reactions. One is acid, another one is aldehyde. Okay, so this is called salicylaldehyde. Okay, salicylaldehyde here. Fine. Yes, or you can call it as 2-hydroxy benzene carbaldehyde. Benzene carbaldehyde. So this is the IUPAC nomenclature you can use. So either reaction for sure you can find in your paper. Okay, and at the same time ಇದನ್ನ even final exam ಅಲ್ಲೂ ಕೇಳ್ತಾರೆ. ನಾ ಹೇಳಿದ್ದೀನಿ ಇದು ಆಲ್ರೆಡಿ ಕ್ವೆಶ್ಚನ್ ಮಾಡಿದ್ದಾರೆ. ವೆರೈಟೀಸ್ ಅಂದ್ರೆ ಅದನ್ನೇ ಸ್ವಲ್ಪ ಮಾಡಿಫೈ ಮಾಡ್ತಾರೆ. ಈಗ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ನಾನು ಇಲ್ಲಿ ಫಿನಾಲ್ ಇಟ್ಕೊಂಡಿದ್ದೀನಿ, ಇನ್ನೊಂದು ಇಲ್ಲಿ ಯಾವುದೋ ಗ್ರೂಪ್ ಕೊಡ್ತಾರೆ, ಮಿಥೈಲ್ ಗ್ರೂಪ್ ಕೊಡ್ತಾರೆ, ಆಗ ಕೇಳ್ತಾರೆ ಇದನ್ನೇ ಇದೇ ಕಂಡೀಷನ್. ಸೊ ನಿಮಗೆ ಫಿನಾಲ್ ರಿಯಾಕ್ಷನ್ ಅಲ್ಲಿ NaOH with chloroform ಬಂದ ಕೂಡಲೇ you should relate it as a Riemann-Tiemann reaction. And the product should be known, that is aldehyde. Ortho position, observe, ಇದು ಯಾವುದು ಫಿನಾಲಿಗೆ ಇದು ಆರ್ಥೋ ಪೊಸಿಷನ್, ಇಲ್ಲೂ ಫಿನಾಲ್ OH ಗ್ರೂಪಿಗೆ ಇದು ಆರ್ಥೋ ಪೊಸಿಷನ್. ಆರ್ಥೋ ಪೊಸಿಷನ್ ಅಲ್ಲಿ ಇಲೆಕ್ಟ್ರೋಫಿಲಿಕ್ ಸಬ್ಸ್ಟಿಟ್ಯೂಷನ್ ಆಗುತ್ತೆ. ಎರಡೂನು ಸಬ್ಸ್ಟಿಟ್ಯೂಷನ್ ಇಲೆಕ್ಟ್ರೋಫಿಲಿಕ್ ಆರೋಮ್ಯಾಟಿಕ್ ರಿಯಾಕ್ಷನ್ಸ್ ಇಲ್ಲೂನು ಆರ್ಥೋ ಪೊಸಿಷನ್ ಅಲ್ಲಿ ಸಬ್ಸ್ಟಿಟ್ಯೂಟ್ ಆಗ್ತಾ ಇದೆ. ಫೈನ್. ಸೊ ಕ್ಲೋರೋಫಾರ್ಮ್ ಫಿನಾಲ್ ಜೊತೆ gives you salicylaldehyde, ಫಿನಾಲ್ ಜೊತೆ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ gives you salicylic acid. ಸೊ ಅದನ್ನ ಲಿಂಕ್ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ, so that you can solve such questions. So this is from the phenols. Now we will take the last reaction, that is from the ether. Onde named reaction irodu, and that is also almost like sure question you can expect. So here I'll go for ether. Only one reaction, that is nothing but Williamson's ether synthesis. Okay, Williamson synthesis or Williamson's ether synthesis. Ethers part bandaga idondu almost sure question. What is this ether synthesis? You know, ethers are represented by a formula ROR dash, alva. So we need these two andre. How we can understand that? Let us take a alkyl halide. Okay, and I need to take another OR dash group should be there. So that is nothing but I'll take it from sodium alkoxide. Alcohol acidic, alcohol chapter allu odidira, acidic nature of alcohol. From alcohol H plus if you replace by reactive metal sodium, you call it as alkoxide. So the reagents required for Williamson's ether synthesis is alkyl halide and an alkoxide. So again you can see a sodium metal here, halogen here. Generally they react to form a compound NaX here. What is left? Left is ROR dash plus NaX. So you got the ether. That's all. And here I can understand this is negative. This will substitute. So this is nothing but you have substitution again. Nucleophilic. Understand the reaction also. So you have a nucleophilic substitution here. Alkoxide reacts with alkyl halide to form what? That is ether. Okay. So I can basically say in alcohols Lucas test is important. In phenols you can find Riemann-Tiemann and Kolbe's reaction, and in ethers only one named reaction, that is Williamson's ether synthesis. Okay, like compared to other previous chapter, less number of reactions here. Yes, now let us see how the questions will be asked. Like MCQs bandaga enirbodu. First of all nan helidini, there should be a little effort to and like remember the reagents. Fine. Yes, one or two example I'll take here. Observe the things carefully. Okay. Now, fine. I told. So look at the substrate. I have a phenol here. I may go with a substituent at this position, that is methyl. I'll write over the arrow mark. We have carbon dioxide. Before that, I'll write sodium hydroxide. Okay, second condition is carbon dioxide, followed by third condition is acidification. Okay. So you should not bother about this group now. This is not reactive to any of this. ಈಗ ಏನ್ ಮಾಡ್ಬೇಕಿಲ್ಲಿ? ಓಕೆ, ಎಸ್, ಫೈನ್. ಈಗ ಏನ್ ಮಾಡ್ಬೇಕು? ಇದು ಈ ರಿಯೇಜೆಂಟ್ಸ್ ಮಾತ್ರ ನೋಡ್ಬೇಕು. NaOH, CO2. ಸೊ ಫಿನಾಲ್ ಇದೆ, NaOH ಇದೆ, ಸೋಡಿಯಂ ಫಿನಾಕ್ಸೈಡ್ ಆಗುತ್ತೆ. ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಇದ್ದಾಗ ಏನಾಗುತ್ತೆ? ಸ್ಯಾಲಿಸಿಲಿಕ್ ಆಸಿಡ್ ಆಗುತ್ತೆ ಅನ್ನೋದನ್ನ ನೀವು ನೆನ್ಪಿಟ್ಕೋಬೇಕು. ಓಕೆ, ಸೊ ಲೆಟ್ ಅಸ್ ಟೇಕ್ ಹಿಯರ್ OH. ಆರ್ಥೋ ಪೊಸಿಷನ್ ಅಲ್ಲಿ ಕಾರ್ಬಾಕ್ಸಿಲಿಕ್ ಗ್ರೂಪ್ ಬರುತ್ತೆ. ಇದು CH3 ಹಾಗೆ ಇರುತ್ತೆ. ಲೆಟ್ ಮಿ ನಾಟ್ ವರಿ ಅಬೌಟ್ ಇಟ್. ಓಕೆ, ಹಾಗೆ. ಅಥವಾ ಈಗ ಐ ಕ್ಯಾನ್ ಟೇಕ್ ಹಿಯರ್, ಲೆಟ್ ಮಿ ಟೇಕ್ ಮಿಥೈಲ್ ಕ್ಲೋರೈಡ್, ಲೆಟ್ ಮಿ ಟೇಕ್ ಸೋಡಿಯಂ ಮೆಥಾಕ್ಸೈಡ್. ಓಕೆ, ಸೋಡಿಯಂ ಮೆಥಾಕ್ಸೈಡ್ ಇಲ್ಲಿ. Look at it. I may not write in this way. ಈ ತರ ಬರೆಯಲ್ಲ ನಾನು. ಡಿಫ್ರೆಂಟ್ ಚೇಂಜ್ ಮಾಡಿ ಬರಿತೀನಿ. ಬಟ್ ನಿಮಗೆ ಆಲ್ಕೈಲ್ ಹ್ಯಾಲೈಡ್ ಇದೆ, ಆಲ್ಕಾಕ್ಸೈಡ್ ಇದೆ. ಎರಡನ್ನ ನೋಡಿದ ಕೂಡಲೇ ರಿಯಾಕ್ಷನ್ ನೆನಪಾಗಬೇಕು. Na ತೆಗೀರಿಲಿಂದ Cl ತೆಗೀರಿಲಿಂದ ಇದೆರಡನ್ನ ಕ್ಲಬ್ ಮಾಡಿ. ಸೊ ವಾಟ್ ಯು ವಿಲ್ ಗೆಟ್? CH3 OCH3 ಮೆಥಾಕ್ಸಿ ಮೀಥೇನ್. ವಿಲಿಯಮ್ಸನ್ಸ್ ಇದು ಬಂದ್ಬಿಟ್ಟು ಕೋಲ್ ಬೇಸ್ ರಿಯಾಕ್ಷನ್. ಸೊ ಹೀಗಿರುತ್ತೆ ಪ್ರಾಡಕ್ಟ್ ಐಡೆಂಟಿಫೈ ಮಾಡೋದು ಅಥವಾ ನೇಮಿಂಗ್ ದ ರಿಯಾಕ್ಷನ್ ವಿಲ್ ಬಿ ದೇರ್. ಫೈನ್. ಓಕೆ, now let us go to the next chapter. What we can take in next chapter? There are comparatively more number of reactions in the next chapter. Aldehydes, ketones bandaga ಸ್ವಲ್ಪ ದೊಡ್ಡ chapter. Reactions are all simple and they are also very, very, very like important, I can say. Fine. Yes, 1 minute. Fine. So let us list it out. Hope you are all listing it so that it will be easy for you. Aldehydes, ketones and carboxylic acid. Yeah, ಬೇಸಿಕಲಿ ಫಸ್ಟ್ ಅಲ್ಲಿ ಏನಂದ್ರೆ ಆಲ್ಡಿಹೈಡ್ಸ್ ಅಲ್ಲಿ ತುಂಬಾ ಇದೆ ನಿಮಗೆ ಪ್ರಿಪರೇಷನ್ ಮೆಥಡ್ಸ್ ತಗೊಂಡಾಗ. ಅದೇ ಕೀಟೋನ್ಸ್ ಬಂದ ಪ್ರಿಪರೇಷನ್ ಸ್ವಲ್ಪ ಕಂಪಾರಿಟಿವ್ಲಿ ಲೆಸ್ ಇದೆ, ಬಟ್ ರಿಯಾಕ್ಷನ್ಸ್ ಅಂತ ಬಂದಾಗ ಎರಡರದ್ದು ಕಾಮನ್ ಇದೆ. ಹಾಗಾಗಿ ದೋಸ್ ಯು ಕ್ಯಾನ್ ಟೇಕ್ ಕಾಮನ್. ಸೊ ಹಿಯರ್ ಫಸ್ಟ್ ಐಲ್ ಡೀಲ್ ವಿತ್ ದ ಆಲ್ಡಿಹೈಡ್ಸ್. ವಿಲ್ ಬಿ ರಿಮೆಂಬರಿಂಗ್ ಒಂದು ಫಸ್ಟ್ ರಿಯಾಕ್ಷನ್ ಬಂದ್ ಬರೋದಿಲ್ಲಿ ರೋಸನ್ಮಂಡ್ ಕೇಳ್ತಾರೆ. ಸೊ ಯು ಟರ್ನ್ ದ ಪೇಜಸ್ ಆಫ್ ಲೈಕ್ ಕಾಂಪಿಟೇಟಿವ್ ಎಕ್ಸಾಮ್ಸ್ ಇದ್ದೇ ಇದೆ, ಐದರ್ ಮ್ಯಾಚ್ ದ ಕಾಲಮ್ ತರ ಕೇಳ್ತಾರೆ ಈ ಚಾಪ್ಟರ್ ಅಲ್ಲಿ ಒಂದಷ್ಟು ರಿಯಾಕ್ಷನ್ಸ್ ನ. ಓಕೆ, ರೋಸನ್ಮಂಡ್ ರಿಡಕ್ಷನ್. ಓಕೆ, ಈ ರೋಸನ್ಮಂಡ್ ರಿಡಕ್ಷನ್ ಏನಪ್ಪಾ, ಹೇಗ್ ನೆನ್ಪಿಟ್ಕೊಳ್ಳೋದು? ರಿಯಾಕ್ಟೆಂಟ್ ಗೊತ್ತಿರಬೇಕು, ಆಲ್ಡಿಹೈಡ್ ಪ್ರಿಪರೇಷನ್ ಆಗ್ತಿರ್ಬೇಕು. ಈಗ ಡೈರೆಕ್ಟ್ ಆಗಿ ನೀವು ಬೋರ್ಡ್ ಅಲ್ಲಿ ಕೇಳಿದಾಗ ಎಕ್ಸಾಮ್ ನೇಮ್ ಹೇಳ್ಬಿಟ್ಟೆ ಹೇಳ್ತಾರೆ. ಅದೇ ಕಾಂಪಿಟೇಟಿವ್ ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ ಸ್ವಲ್ಪ ಕ್ಲೂ ಇರುತ್ತೆ, ಯಾಕಂದ್ರೆ ಕ್ವಶನ್ ಇರುತ್ತೆ, ಸ್ವಲ್ಪ ರಿಲೇಟ್ ಮಾಡಕ್ಕಾಗತ್ತೆ. ಸೊ ಅದೇ ಆಪ್ಷನ್ ನಿಮಗೆ ಲೈಕ್ MCQ ಬಂದಾಗ ಈಗ ನಿಮ್ ಬೋರ್ಡ್ ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ ಅದೊಂದು ಯೂಸ್, ಒಂದ್ ಸ್ವಲ್ಪ ಹಿಂಟ್ ಇರುತ್ತೆ ಅಲ್ಲಲ್ಲೇ ನಿಮಗೆ. ಸೊ ಅದನ್ನ ನೋಡ್ಕೊಂಡಾದ್ರೂ ವರ್ಕ್ ಮಾಡ್ಬೋದು. ಬಟ್ MCQs ನಿಮಗೆ ಏನೋ ಬೋರ್ಡ್ ಅಲ್ಲಿ ಈಸಿ ಆಗುತ್ತೆ ಅಂತ ತುಂಬಾ ಅನ್ಕೋಬಾರ್ದು, ಯಾಕಂದ್ರೆ ಕರೆಕ್ಟ್ ಆಗಿ ನಾಲೆಜ್ ಇರ್ಬೇಕು, ಇಲ್ಲ ಅಂದ್ರೂ MCQs ಮಾಡಕ್ಕಾಗಲ್ಲ. ಸೊ ಇಲ್ಲಿ ರೋಸನ್ಮಂಡ್ ರಿಡಕ್ಷನ್ ಅಲ್ಲಿ ಯು ಕ್ಯಾನ್ ಸಿ ಆಸಿಡ್ ಹ್ಯಾಲೈಡ್ಸ್ ಯೂಸ್ ಮಾಡ್ತೀವಿ, ಬೆನ್ಸಾಯಿಲ್ ಕ್ಲೋರೈಡ್, ಇದು ನೆನಪಿರ್ಬೇಕು. ಇಲ್ಲಿ ನಿಮ್ಗೆ NCERT ನಲ್ಲಿ ಎಕ್ಸಾಂಪಲ್ ತಗೊಂಡಿರ್ಬೋದು, ಯಾವ್ದಾದ್ರು ಚೇಂಜ್ ಮಾಡಬಹುದು ಈ ಗ್ರೂಪ್ ನ. ನಾನು ಸಿಂಪಲ್ RCOCl ಅಂತಾನು ಬರಿಬಹುದು, ಇದನ್ನ RCOCl ಅಂತಾನು ಬರಿಬಹುದು. ಏನಿದೆ ರಿಯಾಕ್ಷನ್ ಇಲ್ಲಿ? ನಮ್ಗೆ ಇದನ್ನ ಆಲ್ಡಿಹೈಡ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಮಾಡ್ತಾ ಇದೀವಿ. ಅಬ್ಸರ್ವ್ ದ ಪ್ರಾಡಕ್ಟ್, ದ CHO ಇರುತ್ತೆ ಇಲ್ಲಿ ಆಲ್ಡಿಹೈಡ್. Cl ತೆಗೆದು ಹೈಡ್ರೋಜನ್ ಆಗ್ತಾ ಇದೀನಿ ಅಂದ್ರೆ ಹೈಡ್ರೋಜಿನೇಷನ್ ಮಾಡ್ತಾ ಇದೀನಿ ಅಲ್ವಾ? ಬಟ್ ಹೈಡ್ರೋಜಿನೇಷನ್ ಮಾಡಿದಾಗ ಈ ಗ್ರೂಪ್ ಏನು ಚೇಂಜ್ ಆಗಬಾರದು. ಹಾಗಾಗಿ ನಾನು ರಿಸ್ಟ್ರಿಕ್ಟೆಡ್ ಹೈಡ್ರೋಜಿನೇಷನ್ ಮಾಡಬೇಕು. ರಿಡಕ್ಷನ್ ಕಂಪ್ಲೀಟ್ ಆಗಿ ಮಾಡುವಾಗಿಲ್ಲ. ಓಕೆ, ಹಾಫ್ ರಿಡಕ್ಷನ್ ಅಂದ್ರೆ ಕಂಟ್ರೋಲ್ಡ್ ರಿಡಕ್ಷನ್ ಅಂತ ಕರಿತೀವಿ. ದಟ್ಸ್ ವೈ ಹೈಡ್ರೋಜಿನೇಷನ್ ಜನರಲಿ ನಾವು ಪೆಲಾಡಿಯಂ ತಗೋತೀವಿ, ಪ್ಲಾಟಿನಂ, ಪೆಲಾಡಿಯಂ, ನಿಕ್ಕೆಲ್ ಎಲ್ಲ ತಗೋತೀವಿ, ಆಗ ಕಂಪ್ಲೀಟ್ ರಿಡಕ್ಷನ್ ಆಗುತ್ತೆ. ಬಟ್ ಇಲ್ಲಿ ಅದನ್ನ ಪಾಯ್ಸನ್ಡ್ ಕ್ಯಾಟಲಿಸ್ಟ್ ಆಗಿ ಮಾಡ್ತೀವಿ, ಪಾಯ್ಸನಿಂಗ್ ಮಾಡ್ತೀವಿ ಬೇರಿಯಂ ಸಲ್ಫೇಟ್ ಕಂಪೌಂಡ್ಸ್ ತಗೊಂಡು. ಓಕೆ, ಸೊ ಇದು ಮಾಡಿದಾಗ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಆಫ್ ಕ್ಯಾಟಲಿಟಿಕ್ ರಿಡಕ್ಷನ್ ವಿಲ್ ಹ್ಯಾಪನ್. ಸೊ ಆಸಿಡ್ ಕ್ಲೋರೈಡ್ ನಿಮ್ಗೆ ಏನಾಗುತ್ತೆ? ಆಲ್ಡಿಹೈಡ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ. ಓಕೆ, ಸೊ ಎನಿ ಆಸಿಡ್ ಕ್ಲೋರೈಡ್ ಈ ರಿಯಾಕ್ಷನ್ ನಾನು ಸಿಂಪಲ್ ಆಗಿ ಇವನ್ ಐ ಕ್ಯಾನ್ ರೈಟ್ ಇಟ್ ಆಸ್ RCOCl with hydrogen gets converted into palladium with barium sulfate ಅಂತ ಆದಾಗ you can have RCHO. Okay, so this is how you can remember the reaction. Second one, we have Stephen's reaction. ಇದು aldehyde preparation, Stephen's reaction ಇದೆ. ಏನು Stephen's reaction? RCN, alkyl cyanide use ಮಾಡಬೇಕು cyanide okay ಮತ್ತೆ hydrogenation but that is reduction reducing agent ಯಾವ್ದು ಯೂಸ್ ಮಾಡ್ತೀವಿ stannous chloride with HCl use ಮಾಡ್ತೀವಿ so stannous chloride with HCl will liberate the hydrogen and converts the cyanide into imine group ಆಗುತ್ತೆ ಈಗ RCHNH ಆಗುತ್ತೆ first understand this will release the hydrogen adds across the double bond here that is one of the pi bond will be cleaved this is intermediate ಇದನ್ನು ಕೇಳ್ತಾರೆ imine formation ಈ imine formation further if you hydrolyze it okay this bond will be cleaved here you can easily understand the hydrolysis like this H2O I can add forms what RCHO and byproduct will be NH3 so aldehyde so alkyl cyanide or nitrile we also call this group as nitrile will get converted into aldehyde preparation fine coming to the next one etarts reaction ಅಂತ ಇದೆ etarts reaction ಇದು ತುಂಬಾ important ಇಲ್ಲಿ especially we are looking into aromatic benzaldehyde ಬೇಕು ಆವಾಗ we'll go for etarts reaction ಅಥವಾ we will go for Guttman-Koch reaction okay what is this etarts reaction here we can bring in two reactions simultaneously but ಒಂದು work ಮಾಡೋಣ first I need to have toluene ಇದು start ಆಗೋದು toluene ಇಂದ so you need to have a toluene or any alkyl benzene ಅಂತ ಅಂತೀವಿ but generally toluene ತಗೊಂಡು work ಮಾಡ್ತೀವಿ because easy to understand then you need to have a chromyl chloride okay so chromyl chloride first ಏನಾಗುತ್ತೆ we need a non-polar solvent CS2 so you will get an intermediate chromium complex here that is CH then we have OCrOH here followed by yes Cl we will be having ice so this is an intermediate you will get here OCr exactly OCr OH with the Cl2 fine Cl will be having one of that correct so this is an intermediate then followed by if you go for hydrolysis this intermediate is also important so you will get here benzaldehyde okay either reaction you can also carry out with the help of what another oxidizing agent that is chromic anhydride so chromic anhydride ತಗೊಂಡಾಗ ಏನಾಗುತ್ತೆ ಸ್ವಲ್ಪ complex will differ but product will remain same ಈಗ ಇದನ್ನ ನೀಟಾಗಿ ಅಬ್ಸರ್ವ್ ಮಾಡಿ chromyl chloride test on ಮಾಡ್ತೀರಾ lab ಅಲ್ಲಿ but that is different for halogen but here chromyl chloride you are taking this is a oxidizing agent ಈ CH3 group observe it is changing as what CHO that means you have added oxygen so this is oxidizing agent so this reaction is a oxidation of benzene okay that is toluene benzene side chain alkyl group ಏನಿದೆ ಅದನ್ನ ಆಕ್ಸಿಡೈಸ್ ಮಾಡ್ತೀರಾ ಇದರ reaction ಅಲ್ಲಿ if you observe Cl is replaced hydrogen cyanide ಏನಾಗಿದೆ ಇಲ್ಲಿ this group unsaturation ತೆಗೆದು you are adding here like hydrogen so these two reactions are reductions ಇಲ್ಲಿ reaction ಅಂತ ಬರೆದ್ರು ಇದನ್ನು reduction but chromyl chloride reaction ಬಂದು oxidation so sometimes ಅದು ಏನ್ reaction ಅಂತಾನು ನಿಮ್ಮನ್ನ ಟೆಸ್ಟ್ ಮಾಡಬಹುದು okay and the last one is here for preparation of aldehyde I'm speaking about that is we take here also benzene but along with the benzene we will go for carbon monoxide and the other reagent required so as it is a formylation reaction we are talking about so CO with HCl and we will be looking at AlCl3 along with CuCl so these are the catalysts required for this particular reaction whenever we take AlCl3 it will be anhydrous observe the reaction so observe the reaction we get CHO okay so we have carbon monoxide and hydrogen together we will introduce a CHO group at the like hydrogen we replace and we introduce so this aldehyde group is nothing but a formyl group so we call it as formylation also and this particular reaction is proposed by Guttman along with we have Koch so this is called Guttman-Koch reaction ಇದು important so again ಮತ್ತೆ ಹೇಳ್ತಾ ಇದ್ದೀನಿ ನಿಮಗೆ reagents substrate ಗೊತ್ತಿರಬೇಕಾಗತ್ತೆ ಇಲ್ಲಾಂದ್ರೆ ಏನು ಮಾಡಕ್ಕಾಗಲ್ಲ ಈಗ ಬಟ್ ಈ ತರ ಕಾಂಬಿನೇಷನ್ ಕೊಟ್ಟಾಗ ಅಟ್ಲೀಸ್ಟ್ ಪ್ರಾಡಕ್ಟ್ ಗೆಸ್ ಮಾಡೋದು ಕಂಪ್ಯಾರಿಟಿವ್ಲಿ ಸ್ವಲ್ಪ ಈಸಿ ಬಟ್ ಎಲ್ಲ ಹೋಲ್ಸಮ್ ಗೊತ್ತಿದ್ರೆ ಈಸಿ ಆಗುತ್ತೆ ವೆದರ್ ದೇ ಆಸ್ ಫಾರ್ ನೇಮ್ ವೆದರ್ ದೇ ಆಸ್ ಫಾರ್ ಪ್ರಾಡಕ್ಟ್ ಯು ಕ್ಯಾನ್ ಡೂ ಇಟ್ ಈಸಿಲಿ ಹೈಡ್ರೋಜನ್ ಇಸ್ ರಿಪ್ಲೇಸ್ಡ್ ಬೈ CHO ಗ್ರೂಪ್ ಸೊ ಫಾರ್ಮೈಲೇಷನ್ ಆರ್ the name of the reaction is Guttman-Koch reaction. ಎಲ್ಲ ನೀವು ಅಬ್ಸರ್ವ್ ಮಾಡ್ದಂಗೆ ರೋಸನ್ಮನ್ ರಿಡಕ್ಷನ್, ಸ್ಟಿಫನ್ಸ್ ರಿಯಾಕ್ಷನ್, ಈಟಾರ್ಸ್ ರಿಯಾಕ್ಷನ್ ಅಂಡ್ ಗಟಮನ್ ಕಾಚ್ ರಿಯಾಕ್ಷನ್ ನಾಲ್ಕೂನು ಯು ಆರ್ ಪ್ರಿಪೇರಿಂಗ್ aldehyde here. Okay, so for ketones preparation ಗೆ ನಾವು ಫಸ್ಟ್ ಪಾರ್ಟ್ ಅಲ್ಲಿ ನೋಡಿದ್ವಿ ಫ್ರಿಡಲ್ ಕ್ರಾಫ್ಟ್ಸ್ ಅಸೈಲೇಷನ್. ಸೊ ಬೆನ್ಝೀನ್ ತಗೊಂಡು ಫ್ರಿಡಲ್ ಕ್ರಾಫ್ಟ್ಸ್ ಅಸೈಲೇಷನ್ ಮಾಡಿದ್ರೆ ನಿಮಗೆ ಅಸಿಟೋಫಿನೋನ್ ಬರುತ್ತೆ, ಅದೊಂದೇ ಇರೋದು ರಿಯಾಕ್ಷನ್. ಸೊ ಅದು ರಿಪೀಟ್ ರಿಯಾಕ್ಷನ್, ನೀವು ಅದನ್ನ ರಿಲೇಟ್ ಮಾಡ್ಕೋಬಹುದು. ಈಗ ರಿಯಾಕ್ಷನ್ಸ್ ಅಂತ ಬಂದಾಗ ಆಲ್ಡಿಹೈಡ್ಸ್ ಕೀಟೋನ್ಸ್ ಅಲ್ಲಿ there are two important reduction, that is Clemenson and Wolff-Kishner, and there are like two important other reactions are condensation, aldol condensation ಮತ್ತೆ Cannizzaro reaction. So first we will work with both put together. We are taking now aldehydes and ketones here. ಈಗ ಈ ಕಾರ್ಬೋನಿಲ್ ಗ್ರೂಪ್ ಇದ್ದದ್ದು, ಸೊ ಆರ್ ಅಂಡ್ ಹೆಚ್ ಹಾಕಿದ್ರೆ ಆಲ್ಡಿಹೈಡ್ ಆಗುತ್ತೆ, ಆರ್ ಅಂಡ್ ಆರ್ ಡ್ಯಾಶ್ ಹಾಕಿದ್ರೆ ಕೀಟೋನ್ ಆಗುತ್ತೆ, ಅದ್ ನಿಮಗೆ ಗೊತ್ತು. ಸೊ ಈ ಕಾರ್ಬೋನಿಲ್ ಗ್ರೂಪ್ ನ ಐ ಕ್ಯಾನ್ ರೆಡ್ಯೂಸ್ ಟು ವಾಟ್ ಹಿಯರ್, ಲುಕ್ ಎಟ್ ಇಟ್, oxygen ತೆಗೆದು I have put hydrogen. That means addition of hydrogen I did, removal of oxygen I did. So what is that addition of hydrogen or removal of oxygen is called reduction. So both this reaction what I'm talking about is reduction. So I can make it bit visible like this. CO group I can convert into CH2. Here also the same end product is same, but reagents are different. In the first case I'll use zinc amalgam with the concentrated HCl. Okay, so this particular reaction is called Clemenson reduction. Clemenson ಅಥವಾ Wolff-Kishner ಅಲ್ಲಿ end product same, ಆದ್ರೆ reagents ಬೇರೆ ಇರುತ್ತೆ. ಇದು the second reaction is, here we will be using other reagent. What is that? First we will go for hydrazine, NH2 first. Second step we will take KOH with ethylene glycol. Ethylene glycol. ಇದೆರಡು reagent. But product same, but the name of the reaction is Wolff-Kishner reduction. CO group ಇದೇನಾಗ್ತಾ ಇದೆ, just it is getting converted into here CH group ಅಷ್ಟೇ. ಈ ಎರಡು ವ್ಯಾಲೆನ್ಸಿ ಏನ್ ಗ್ರೂಪ್ ಇದೆ ಅದನ್ನ ಹಾಗೆ ಬರಿಬಹುದು. ಎಲ್ಲಿ ಕಾರ್ಬೋನಿಲ್ ಗ್ರೂಪ್ ಇದೆಯಾ ಆಕ್ಸಿಜನ್ ರಿಮೂವ್ ಮಾಡಿ ಹೈಡ್ರೋಜನ್ ಹಾಕಿದ್ರೆ ಆಯ್ತು. ಅಂದ್ರೆ aldehydes or ketones are reducing to alkane. That is a final point you need to remember. Okay, ಇದೆರಡು reaction, named reaction you have. Then comes to other one is aldol condensation. ಏನಿದೆ ಆಲ್ಡಾಲ್, ಇದು ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್. ಇದಕ್ಕೆ ಸ್ಪೆಸಿಫಿಕ್ ಆಗಿ ಸೈಂಟಿಸ್ಟ್ ನೇಮ್ ಏನಿಲ್ಲ. ಓಕೆ, ದಿಸ್ ಇಸ್ ನಾಟ್ ದ ನೇಮ್ ಆಫ್ ಸೈಂಟಿಸ್ಟ್, ಬಟ್ ದ ಪ್ರಾಡಕ್ಟ್ ಫಾರ್ಮ್ಡ್ ಹ್ಯಾಸ್ ಎ ಆಲ್ಕೋಹಾಲ್ ಅಂಡ್ ಆಲ್ಡಿಹೈಡ್ ಇನಿಷಿಯಲಿ, ಅದಕ್ಕೆ ಆಲ್ಡಾಲ್ ಅಂತ ಕರಿತೀವಿ. ಬಟ್ ಇದು ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್. ಯಾವ್ದು ಆಲ್ಡಾಲ್ ಕಂಡೆನ್ಸೇಷನ್ ಆಗುತ್ತೆ? ಐದರ್ ಯು ಹ್ಯಾವ್ ಎ ಆಲ್ಡಿಹೈಡ್ ಆರ್ ಕೀಟೋನ್ ವಿತ್ ಆಲ್ಫಾ ಹೈಡ್ರೋಜನ್. ಇದು ಕಂಡೀಷನ್, ಆಲ್ಫಾ ಹೈಡ್ರೋಜನ್ ಇರಲೇಬೇಕು. ಓಕೆ, ಸೊ ಒಂದ್ ಎಕ್ಸಾಂಪಲ್ ವರ್ಕ್ ಮಾಡ್ತೀನಿ, ಅಷ್ಟು ಟೈಮ್ ಕನ್ಸ್ಟೆಂಟ್ ಇರೋದ್ರಿಂದ ಒಂದ್ ಎಕ್ಸಾಂಪಲ್ ವರ್ಕ್ ಮಾಡ್ತೀನಿ ನಾನು. ಓಕೆ, ಸೊ ವಿ ವಿಲ್ ಟೇಕ್ ಹಿಯರ್ CH3 CHO. ಓಕೆ, ನಾನು CHO ನ ಈ ತರ ಬರೀತೀನಿ, ಅಸಿಟಾಲ್ಡಿಹೈಡ್. ಈಗ ಇನ್ನೊಂದು ಮಾಲಿಕ್ಯೂಲ್, ಸೊ ದಿಸ್ ಇಸ್ ವಾಟ್, ದಿಸ್ ಇಸ್ ಯುವರ್ ಆಲ್ಫಾ ಕಾರ್ಬನ್, ಈ ಹೈಡ್ರೋಜನ್ ಬೇಕು. ಈ ರಿಯಾಕ್ಷನ್ ಮೆಕ್ಯಾನಿಸಮ್ ನೀವು ಡೀಟೇಲ್ ಆಗಿ ಕ್ಲಾಸ್ ಅಲ್ಲಿ ಮಾಡಿರ್ತೀರಾ, ಬಟ್ ನಾನು ಇಲ್ಲಿ ನಿಮಗೆ ಸಿಂಪಲ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಸೊ ಎರಡು ಮಾಲಿಕ್ಯೂಲ್ ನ ಈ ತರ ಬರ್ಕೊಳ್ಳಿ. ಒಂದು ಮಾಲಿಕ್ಯೂಲ್ ಇದು CHO, ಆಕ್ಸಿಜನ್ ಡೈರೆಕ್ಟಿಂಗ್ ಟುವರ್ಡ್ಸ್ ಅನದರ್ ಮಾಲಿಕ್ಯೂಲ್. ಅದರಲ್ಲಿ ಆಲ್ಫಾ ಕಾರ್ಬನ್ ಇದು ಹೈಡ್ರೋಜನ್ಸ್ ಎರಡು ಈ ಕಡೆ ಓರಿಯೆಂಟ್ ಆಗೋಂಗೆ ಮಾಡ್ಬಿಡಿ. ಸೊ ದಟ್ when I club I can remove this water molecule orientation ಮಾಡ್ತಾ ಇದ್ದೀನಿ, so that ನಿಮಗೆ ಈಸಿ ಆಗುತ್ತೆ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಕ್ಕೆ ಅಂತ. We need here dilute NaOH and heating, then only the condensation ಆಗೋದು. ಈಗ ತೆಗ್ದಾಗ ಏನಾಯ್ತು, you will get CH3 CH double bond CH and CHO. So this is condensation product. So any aldehyde or ketone with alpha hydrogen can undergo condensation. ಅದಕ್ಕೆ alpha carbon, alpha hydrogen, this is functional group. Functional group ಪಕ್ಕ ಇರೋ ಕಾರ್ಬನ್ is alpha carbon. ಅದರಿಂದ ಎರಡು ಹೈಡ್ರೋಜನ್ adjacent ಇನ್ನೊಂದು ಸೇಮ್ ಮಾಲಿಕ್ಯೂಲ್, ಅದರದ್ದು ಆಕ್ಸಿಜನ್. You have to remove it as water and club it and write the product. Okay, next reaction is Cannizzaro reaction. ಸೊ ಈ Cannizzaro ರಿಯಾಕ್ಷನ್ ಯಾವ್ದ್ರಿಂದ ನಿಮ್ಗೆ ಸಿಗುತ್ತೆ ಅಂದ್ರೆ ಓನ್ಲಿ ಆಲ್ಡಿಹೈಡ್. ಓಕೆ, ಆಲ್ಡಿಹೈಡ್ ವಿತ್ ಆರ್ ವಿದೌಟ್ ಆಲ್ಫಾ ಹೈಡ್ರೋಜನ್, ಆಲ್ಡಿಹೈಡ್ ವಿದೌಟ್ ಆಲ್ಫಾ ಹೈಡ್ರೋಜನ್. ಆಲ್ಫಾ ಹೈಡ್ರೋಜನ್ ಇದ್ರೆ aldol condensation ಆಗುತ್ತೆ. ವಿತೌಟ್ ಆಲ್ಫಾ ಹೈಡ್ರೋಜನ್ ಅಂದ್ರೆ ಅನಿಸೋರಿ ರಿಯಾಕ್ಷನ್ ಆಗುತ್ತೆ. ವಿತೌಟ್ ಆಲ್ಫಾ ಹೈಡ್ರೋಜನ್ ಅಂದ್ರೆ ನಿಮಗೆ ಫಾರ್ಮಾಲ್ಡಿಹೈಡ್, ನಿಮಗೆ ಗೊತ್ತು ಫಾರ್ಮಾಲ್ಡಿಹೈಡ್ ಅಲ್ಲಿ ಇರೋದೇ ಒಂದು ಕಾರ್ಬನ್, ಫಂಕ್ಷನಲ್ ಗ್ರೂಪ್, ಅದ್ರ ಪಕ್ಕ ಕಾರ್ಬನೇ ಇಲ್ಲ, ಹಾಗಾಗಿ ಹೈಡ್ರೋಜನ್ ಇಲ್ಲ ಅಲ್ಲಿ. ಓಕೆ, ಸೊ ನೋ ಆಲ್ಫಾ ಹೈಡ್ರೋಜನ್. 2 ಮಾಲಿಕ್ಯೂಲ್ ತಗೋಬೇಕು. ಇಲ್ಲಿ ಕಂಡೀಷನ್ ಬಂದ್ಬಿಟ್ಟು ಕಾನ್ಸಂಟ್ರೇಟೆಡ್ ನಿಮಗೆ KOH ಅಥವಾ NaOH ಯೂಸ್ ಮಾಡಬಹುದು. ಒಂದು ಮಾಲಿಕ್ಯೂಲ್ ವಿಲ್ ಗೆಟ್ ಆಕ್ಸಿಡೈಸ್ ಟು its carboxylic group RCOOK. Another molecule illideala innond molecule that will get reduced to what? Alcohol. So aldehyde, this aldehyde gets oxidized to acid, oxidation I wrote. Other one aldehyde here that get reduced to alcohol. This is how you can understand the products. Okay, so this is a Cannizzaro reaction here. Cannizzaro reaction condition andre aldehyde without alpha hydrogen. With alpha hydrogen will always undergo that is aldol condensation. Fine. ಆಮೇಲೆ in this particular chapter you will be having another 2 tests. ತುಂಬಾ important. What are they? Tollens test and the failing test. ಲ್ಯಾಬ್ ಅಲ್ಲೂ ಮಾಡ್ತೀರಾ, ಈವನ್ ಬಯಾಲಜಿ ಲ್ಯಾಬ್ ಅಲ್ಲೂ ಯೂಸ್ ಮಾಡ್ತೀರಾ, ಹಾಗೆ ಕ್ವೆಶ್ಚನ್ಸ್ ಬರೋ ಚಾನ್ಸಸ್ ತುಂಬಾ ಇರುತ್ತೆ. ಸೊ one is like if you take a tolerance test here, so I can just make a quick like chart for you. Another one is failing test. Reagent ಗೊತ್ತಿರಬೇಕು ನಿಮಗೆ. tolerance test ಗೆ silver nitrate with the ammonium hydroxide ಅಥವಾ ಅಮೋನಿಯಾಕಲ್ ಸಿಲ್ವರ್ ನೈಟ್ರೇಟ್ ಅಂತೀವಿ. ಫೇಲಿಂಗ್ ಸೊಲ್ಯೂಷನ್ 2 ತಗೋತೀವಿ, ಒಂದು aqueous copper sulfate, another one is alkaline sodium potassium tartarate, that is called Rochelle salt. Okay, Rochelle salt. These are the 2 like reagents, I can say reagents here. Full reaction bariyalla, you can refer to the book. If I have aldehyde, okay, aldehyde will answer tolerance test here, right? Even failings test. But if I take a ketone, you know ketones, right? Ketones won't answer for tolerance and failings. So question ಹೇಗಿರುತ್ತೆ ಅಂದ್ರೆ, so they'll give you, because aldehyde and ketone ಎರಡೂನು they have a general formula CnH2nO, right? So ಈಗ ನಾನು CnH2nO ಅಂತ ತಗೊಂಡಾಗ you can have propanol and propanone, right? So C3H6O molecular formula ಅಂದ್ರೆ ಇದು CH3 CH2 CHO ನೂ ಆಗ್ಬೋದು, ಅಥವಾ CH3CO CH3 ನೂ ಆಗ್ಬೋದು. ಇದು ಕೀಟೋನ್, ಇದು ಆಲ್ಡಿಹೈಡ್. ಈಗ ಒಂದು ಆರ್ಗ್ಯಾನಿಕ್ ಕಂಪೌಂಡ್ ಇದು ಮಾಲಿಕ್ಯುಲರ್ ಫಾರ್ಮುಲಾ ಇದೆ, ಇದು ಆಲ್ಡಿಹೈಡ್ ಕೀಟೋನಾ ಅಂತ ಹೇಗೆ ಫೈಂಡ್ ಔಟ್ ಮಾಡಬೇಕು ಅಂತಂದ್ರೆ you have to go for tolerance test. Tolerance test is easy. You will get a silver mirror here. ಏನು ಇನ್ಫರೆನ್ಸ್ ಹಿಯರ್, ಕೀಟೋ ಆಲ್ಡಿಹೈಡ್ಸ್ ಅಲ್ಲಿ ಇಲ್ಲಿ ಸಿಲ್ವರ್ ಮಿರರ್ ಫಾರ್ಮ್ ಆಗುತ್ತೆ. ಇಲ್ಲಿ you will get a Cu2O ರೆಡ್ ಪ್ರೆಸಿಪಿಟೇಟ್ ಫಾರ್ಮ್ ಆಗುತ್ತೆ. ದಟ್ ಇಸ್ ದ ರಿಯಾಕ್ಟಿವ್ ಗ್ರೂಪ್ is nothing but copper ion ಅಲ್ಲಿ. ಸೊ ಇದನ್ನ ನಾವು ಈಸಿಯಾಗಿ ಐಡೆಂಟಿಫೈ ಮಾಡಬಹುದು. ಟಾಲೆನ್ಸ್ ಟೆಸ್ಟ್ ಮಾಡಿ ಸಿಲ್ವರ್ ಮಿರರ್ ಫಾರ್ಮ್ ಆರ್ ಆಲ್ಡಿಹೈಡ್, ನೋ ಸಿಲ್ವರ್ ಮಿರರ್ ಮೀನ್ಸ್ ಕೀಟೋನ್. Then if you want to confirm again, you can go for failings. Red precipitate under aldehyde, otherwise it will be a ketone here. Okay, and remember this failing test is not answered by aromatic aldehyde. That condition in pitkobeku. All aliphatic will answer, but aromatic won't answer. Okay, that is another named reaction you have, or just to distinguish aldehydes and ketones. Last one in this chapter is HVZ reaction. This is the only reaction, named reaction for carboxylic acid. That is called Hell-Volhard-Zelensky reaction. Okay, Zelensky reaction. Yes, what is the requirement? Carboxylic acid, again you need to have alpha hydrogen here. This is alpha carbon, alpha hydrogen. Condition is you are going to treat it with halogen, generally chlorination, with the red phosphorus. So this is a reaction condition you can find. Okay, so we will be having one red phosphorus condition along with the halogen. For finally you can go for hydrolysis here. Yes, hydrolysis. What is happening? This alpha hydrogen is replaced by halogen, RCH XCOOH. So HVZ reaction is nothing but you are carrying out halogenation. So ಇಲ್ಲಿ ನೋಡಿ halogen ತಗೊಂಡಿದ್ದೀನಿ, ಆ halogen ಇಲ್ಲಿ ಬರುತ್ತೆ. One of the hydrogen is substituted by halogen. So it is a halogenation reaction, but with the your acid provided with that has a alpha carbon and it has a alpha hydrogen. So you cannot expect this to happen with the formic acid. Acetic acid alagata, higher acid cell provided with they have a alpha hydrogen. They all can answer for this particular, like they can give you the Helvoland-Zelensky reaction. Okay. So illi basically name of the reaction, or you can get product identification. ಮಾಲಿಕ್ಯೂಲ್ ನ ಸ್ವಲ್ಪ ಚೇಂಜ್ ಮಾಡ್ಬೋದು. ನಿಮ್ಗೆ NCERT ನಲ್ಲಿ ಸಿಂಪಲ್ ಅಸಿಟಾಲ್ಡಿಹೈಡ್ ಕೊಟ್ಟಿದ್ರೆ ಅಲ್ಲಿ ಪ್ರೊಪೆನಾಲ್ಡಿಹೈಡ್ ಕೇಳ್ಬೋದು, ಪ್ರೊಪಿಯೋನಾಲ್ಡಿಹೈಡ್ ಕೇಳ್ಬೋದು, ಅಥವಾ ಬ್ಯೂಟಾರಾಲ್ಡಿಹೈಡ್ ಕೇಳ್ಬೋದು. ಸೊ ಸ್ಮಾಲ್ ಚೇಂಜಸ್ ಮಾಡ್ತಾರೆ. ಹಾಗಾಗಿ ಬೇಸಿಕ್ ಸ್ಕೆಲಿಟನ್ ಈಕ್ವೇಶನ್ ನಿಮಗೆ ಗೊತ್ತಿದ್ರೆ ಯು ಕ್ಯಾನ್ ಜಸ್ಟ್ ಈಸಿಲಿ ವರ್ಕ್ ವಿತ್ ಆಲ್ ದಿಸ್ ನೇಮ್ಡ್ ರಿಯಾಕ್ಷನ್ಸ್. ಸೊ ಐ ಕುಡ್ ಬಿ able to complete only 3. You have an amines chapter, but in amines chapter hardly you have countable, only 2. One is your Gabriel thalamide, and in the other reaction is Hoffman bromamide degradation, which is easy actually. So you can go through those two, you can add to the list. Then if you want, again in the amines chapter alli tumba important andre test to distinguish amines. One carbyl amine, primary amines matra answer ಮಾಡುತ್ತೆ. Second one ಬಂದು Hinsberg's reagent ಯೂಸ್ ಮಾಡ್ತೀರಾ primary secondary tertiary amine ನ. ಸೊ ಅಲ್ಲಿ ಪ್ರೈಮರಿ ಸೆಕೆಂಡರಿ ಟರ್ಷರಿ ಬಂದಾಗ ಮೀನಿಂಗ್ ಡಿಫರೆನ್ಸ್ ಆಗುತ್ತೆ. ಅದನ್ನ ಅಬ್ಸರ್ವ್ ಮಾಡಿ. ಪ್ರೀವಿಯಸ್ ಒನ್ ಆಫ್ ದ ಸೆಷನ್ಸ್ ಅಮೈನ್ಸ್ ಬಗ್ಗೆ ನಾವು ಡಿಸ್ಕಸ್ ಮಾಡಿದ್ವಿ. ಅಲ್ಲಿ ಆಲ್ ದಿಸ್ ಇನ್ಫಾರ್ಮೇಶನ್ ಐ ಹ್ಯಾವ್ ಗಿವನ್. ಸೊ ದಟ್ ಕಂಪ್ಲೀಟ್ ಅಮೈನ್ಸ್ ವಿ ಹ್ಯಾವ್ ಡೆಲ್ಟ್ ಒನ್ ಕ್ಲಾಸ್. ಸೊ ಯು ಕ್ಯಾನ್ ರೆಫರ್ ಟು ದಟ್ and you can add those reaction to your list. So I think that will be very useful. You list to reagent to end product form ಆಗುತ್ತೆ with the name. If you have every day, like as I told, like how you learnt your rhymes or how you learnt your formulas in maths. So ಹೋಗ್ತಾ ಬರ್ತಾ ಒಂದೆರಡು ಸತಿ ನೋಡಿದ್ರೆ ರಿಜಿಸ್ಟರ್ ಆಗ್ತಾ ಇರುತ್ತೆ. Try to recall every day. I think you can have good number of questions from all these named reactions. Okay, I think with this I will stop today's session. If you have any doubts, you can definitely ask. Yes sir.
Thank you, madam.
Thank you sir. Any
So-
questions from the student side?
ಸೊ ಚಾಟ್ ಬಾಕ್ಸ್ ಅಲ್ಲಿ ಯಾವುದೇ ಕ್ವಶನ್ಸ್ ಇಲ್ಲ ಮೇಡಮ್.
Okay,
ಸೊ ಲೇಟರ್ ಆನ್ ಕಳಿಸಿದ್ರೆ ನಾನು ನಿಮ್ಗೆ
sure sir.
ಸ್ವಲ್ಪ ಈಗ ಎಕ್ಸಾಮ್ ಹತ್ರ
Exam.
ಇರೋದ್ರಿಂದ ಮಕ್ಕಳು ಜಾಸ್ತಿ ಇಂಟ್ರೆಸ್ಟ್ ಆಗಲ್ಲ ಒಂದು.
ಹೌದು ಡೆಫಿನೆಟ್ಲಿ. ಬಟ್
So,
ಆಮೇಲೆ
ಏನಾದ್ರು-
ನೋಡಿದ್ರೆ ಯೂಸ್ ಆಗುತ್ತೆ ಅವ್ರಿಗೆ ಅಷ್ಟೇ.
ಎಸ್ ಮ್ಯಾಮ್. ಈಗ ಆಲ್ರೆಡಿ ಫಸ್ಟ್ PU ಮಕ್ಕಳುಗಳಿಗೆ ಎಕ್ಸಾಮ್
Exam
ಶುರು
ಆಗ್ತಾ
ಆಗಿದೆ
ಇದೆ.
17th ಇಂದ.
ಹೌದು.
ಸೆಕೆಂಡ್ PU ಮಕ್ಕಳುಗಳಿಗೆ ಐ ಥಿಂಕ್ ಫಸ್ಟ್ ವೀಕ್ ಶುರು ಆಗುತ್ತೆ ಮೇಡಮ್.
ಹೌದು ಮಾರ್ಚ್.
ಹಾಗಾಗಿ ಅವ್ರು ಏನಾದ್ರು ಕಳಿಸಿದ್ರೆ ಐ ಲೇಟರ್ ಆನ್ ಸೆಂಡ್ ಥ್ರೂ ಮೇಲ್.
Sure sir. Okay sir then.
Anyway, thank you ma'am.
Thank you sir.
So delivered a wonderful informative talk on today's topic. Is ಮಹತ್ತರವಾದ ವಿಷಯಗಳನ್ನ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ತಿಳಿಸ್ಕೊಟ್ಟಿದ್ದಾರೆ ಮೇಡಮ್. ಸೊ ನನ್ನ ವೈಯಕ್ತಿಕವಾಗಿ ಹಾಗೂ ನಮ್ಮ ಅಕಾಡೆಮಿ ಪರವಾಗಿ once again ಧನ್ಯವಾದಗಳು ಮೇಡಮ್.
Thank you so much sir for the opportunities. Yes sir. Okay sir then.
ಹಾಗೂ ನಮ್ಮ ಅಕಾಡೆಮಿ ಅಧ್ಯಕ್ಷರಾದಂತ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ ಕೂಡ ನನ್ನ ಧನ್ಯವಾದಗಳನ್ನ ತಿಳಿಸುತ್ತಾ, ನಮ್ಮ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನು ಆಯೋಜಿಸಿ ಪ್ರತಿನಿತ್ಯವೂ ಸಲಹೆಗಳ ಸೂಚನೆಗಳನ್ನು ನೀಡುತ್ತಾ ಬಂದಿರುವ ನಮ್ಮ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದಂತ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳು ಸರ್. ಹಾಗೂ ಇಂದು ಹಾಜರಾಗಿದ್ದ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿಗಳು ಹಾಗೂ ನನ್ನ ಸಹೋದ್ಯೋಗಿ ಮಿತ್ರರು ಹಾಗೂ ವೆಬೆಕ್ಸ್ ವೃಂದದವರಿಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳು ತಿಳಿಸುತ್ತಾ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮವನ್ನು ಮುಗಿಸುತ್ತಿದ್ದೇವೆ. Once again, thank you all. Thank you ma'am.
Thank you sir.
Thank you.